are going to start our first session. And so GP Singh, PhD, I joined and he will be into this like to talk. I request Professor Singh Sahab to give a brief introduction of the question and before he starts his talk, please.
Thank you, Professor Singh sir.
Okay, sir.
Good morning.
Good morning. Good morning.
Before Professor Singh will start his talk, I will be brief about his work, what he has done. He is a professor in physics at Biodiversity University, and he is working on the influence of climate change on the total Southeast Asia. He is working with the collaboration with the South Korea University. He is a very widely visited across 20 countries. Yes, and why we keep this talk because he is making modeling on the forecast of the climate. The whole India just has been brief. He is what his main work is. He takes the rainfall and temperature of the area last 100 years again and takes the whole India data and shows it with the actual things happens and what will be the right way in the future can be happen and what will be its influence on different things. Why is having a base for this environmental because geophysics department is Banaras Hindu University mostly working on these lines. The geospatial inside Earth climate, outside Earth climate, space climate. So Professor Singh has done very nice work and it is our pleasure to have you Professor Singh today and we will end it by listening your lecture. I hope you will also enjoy, but I will say that you ask the question after completion of this talk. Thank you. Thank you, Professor Singh. Please, you can now start your lecture.
Sir, your mic is not on. Sir, your mic is not on. Mute. Please unmute it.
Hello. Hello.
Yeah, it's audible now.
It is audible?
Yeah, it's audible now. Please go ahead, sir.
Oh, are you able to see my slide? Hello.
Yeah.
Can you see my slide?
Is visible to you. Yes, it is visible. I think.
Thank you very much. So today I have selected the topic for my presentation: projected changes in temperature, precipitation, and extreme events over different parts of India and East Asia and their teleconnection with a high resolution atmospheric general circulation model (AGCM) during 21st century. So, as you are aware, nowadays many people are working. I can say the climate change is a topic of very burning topic of research. So there has been a lot of discussion in the media as well as in the political about the climate change in connection with the greenhouse effect and the expected global warming. So, in fact, no other environmental issue has garnered more attention than the climate change and associated debate over the global warming. So this issue has been topic of countless hearing over the past several years, and most of which have been essentially economy in focus, although some have dealt directly with the climate change. Even the man on the street is aware about the consequence of the calamities associated with the continued global warming. So the list is unending, like a few of them are rise in sea level rising, hot spell in summer, drought and flood condition, hurricanes, etc., etc. So I can say that whenever some unusual bad weather condition occur, the blame directly goes to the global warming because it had happened because of the global warming. So now I want to give you an example. So actually in 2002, there was a very heavy drought condition prevail over the long period of time over the Europe. And the media, BBC people, media came to the International Center for Theoretical Physics, that is ICTP in Italy, and they have asked what is the reason behind the drought of 2002. So the scientists told that the reason is only global warming. And the next year in 2003, there was a very heavy flood condition occur over the large part of the Europe. And then again, same media people came and asked, what is the reason behind the high flood condition over the European region? Again, scientists uh, told them that it is because of the global warming. So that is why I'm telling that if some unusual event occur and that is directly related to the global warming, it is because of global warming. So actually the idea of the global warming has come in there was a very severe drought condition in 1988 in USA, and it was the worst drought condition in the and it attracted full media attention. And many people thought that this was the proof that the man-made greenhouse effect and finally brought the expected global warming. In fact, during 1980s, one temperature record break the another temperature record. So same thing happened again in 1990. Then the question now arises, has global warming arrived? So then we can come to the conclusion that the topic of the climate change is just like a puzzle, and there are many different pieces like ocean, atmosphere, ecosystem, polar ice, natural and human influence. Then scientists have been working on this puzzle for more than 3-4 decades, and while there are still gaps in our knowledge, so most experts feel we have the puzzle that is incomplete enough to show the human activities are having an adverse effect on our planet. So this talk looks at many of those puzzle pieces, the evidence behind them, and the conclusion we can draw from them. So as you know, in the atmosphere, there are different type of gases are present, like a nitrous, some of the fixed gases and some of the variable gases, like nitrogen, oxygen, xenon, and there are many gases, but these include here about more than 99%. And less than 1% is the carbon dioxide, water vapor, and ozone. So these are comes under the category of greenhouse gases. So these variable gases are mostly responsible for the global warming. So as you know, the carbon dioxide comes from the variety of sources. For example, plants take up carbon dioxide in the air to make wood, stem, and leaves, and then release it back into the air. When the leaves fall or the plant die, the concern today is that fossil fuel use is putting huge amount of carbon dioxide in the atmosphere, at the rate faster than the climate system can adapt. So in addition, the warming that is resulting from the carbon dioxide and other greenhouse gases also has effect on the increasing the evaporation. Or this adds the water vapor into the atmosphere as well. So water vapor is the most important gas in the natural greenhouse effect that contribute very huge amount of absorption of the incoming solar outgoing long wave radiation. So the first, it was realized that it is better to take the actual observation of the carbon dioxide. So first time, Sir Gilbert Charles Keeling, Dr. Charles Keeling, he was the first person to start the real observation of the carbon dioxide. Actually, he has measured he has put his laboratory at the Mauna Loa, that is in Hawaii, and data were collected at the Mauna Loa Observatory, and this is near about 3.4 kilometer above the mean sea level, and above sea level on the northern slope of the Earth's largest volcano. That is away from the dense vegetation or human population, and that might skew the measurement. And the bare volcanic landscape surrounding this observatory is shown here. So this will give the very proper observation of the carbon dioxide. And he took the data from 1958 to 2008, and he found that the concentration of the carbon dioxide in the atmosphere is increasing continuously. If you look in 1950, it was very close to 310 ppm, but up to 2005, it had reached more than 380 ppm. And actually, the main emission of the carbon dioxide had taken place during the time of James Watt. He had searched the steam engine, so we call it as a industrial revolution has come after that. So if you see this picture, on the x-axis, that is the year, and the y-axis is the concentration of the carbon dioxide. If you look from 1700 to 2050, so this dotted line show the time during the time of James Watt. At that time, the concentration of the carbon dioxide was approximately 275 ppm in the atmosphere. But after that, Dr. Sir Charles Keeling observation that is shown by the red line. So you can show this is the steep rising in the concentration of the carbon dioxide. Thus, we can see, you can say that carbon dioxide is the main culprit who has contributed in the global warming. And some of the model run, and it has been also found by the IPCC, Intergovernmental Panel on Climate Change, IPCC report. There are different scenarios, and IPCC also reported that he has given the relative change from 1980 to 1999 and 2090 to 2099. How much relative change takes place during the 100 years? So you can see here, B1 scenario, it was giving the increase in temperature 1.8 degree, B2 2.4 degree, 2.8 degree, 2.4 degree, 3.4 degree, 4 degree. So what you can see here, mostly in all the scenario, temperature is increasing. So we can say that global warming is taking place, and its impact is that the sea level is rising. Using the same scenario, it has been found that the sea level was rising from 0.18 to 0.38 meter. If you look in all the scenario, some of the scenario is giving higher, let's say up to 0.59 meter. So you can say that increase in sea level rising is from 0.18 to 0.59 meter. I will tell more detail later. So here I would like to, this is our main content. So global warming is no longer an issue for debating. because we have seen so many results which have clearly show the global warming is taking place. Then what changes the climate? Should we care? And then I will come to the observed climate change over India, and then I will come to the projected change in temperature and precipitation using the GME model, and how sure are scientists about the global warming, and what next, what can we do, what will be our contribution in reducing the global warming. So you know that global warming takes place because of the various reasons. If there is a change in the output energy of the sun, it has also impact on the incoming solar radiation of the Earth's surface. So it has also impact on the temperature. Earth orbit, as you know, Earth rotates in orbit. It has also some impact on the atmosphere. You know that repetitive cycle in the Earth orbit that occur over tens of thousands of years can influence the angle and timing of the sunlight. Drifting of continent. So you know that in distant past, drifting continent make a big difference in the climate over millions of years by changing the ice cap at the pole and by altering the ocean current, which transport heat and cold throughout the ocean depth. And then volcanic eruption. You know that huge volcanic eruption can cool the earth by injecting ash and tiny particle into the atmosphere. And last one is the greenhouse gases. So if you look this one from sun output, earth orbit, drifting of continent and volcanic eruption, these are the natural changes where we don't have any control on these changes. And last one is the greenhouse gases. We can say it is the anthropogenic changes. So as I told you, in the atmosphere, there are different greenhouse gases are present like nitrous oxide, carbon dioxide, methane, water vapor, and sulfur hexyl chloride. So these gases are present in the atmosphere. So what happens? When the incoming solar radiation reach at the surface of the Earth absorbs this energy, and then after absorbing this energy, it re-radiates in the atmosphere in the form of long-wave radiation. So this radiation, and as I told you, that different greenhouse gases are present in the atmosphere. This don't have any impact on the sun radiation, which is coming, or short wave radiation. But when Earth emits the radiation in the long wave, these gases are very good absorber of the long wave radiation, and then it re-radiates again to the Earth's surface. So it looks like an umbrella. So it allows sun radiation to reach at the surface of the Earth, but doesn't allow long wave radiation go to the atmosphere. So this way, the atmosphere now become heated. So then question arises, this type of global warming, it takes place this time a few decades or it was back also. So here I want to show you the picture of 800,000 years back. So upper one, this blue line show the variation in the concentration of the carbon dioxide, and the lower one show the variation in the concentration of variation in the temperature variation. So if you look upper one, so you can find that in the past there was a huge variation in the carbon dioxide, but that variation is not cross the limit that is shown by the dotted line. And here temperature is also follow exactly the variation in the carbon dioxide. So we can say that carbon dioxide actually the main cause, it has impact on the variation in the temperature. So, but during the recent few years, it has been found that concentration of the carbon dioxide has reached up to 380 ppm, and the temperature also had reached more than 1 degree has been rising. So this way, the carbon dioxide and temperature has a very close relation. Then, is it real warming? So for that purpose, we want to show you the entire globe variation in temperature and ocean variation and land variation. So if you look this one, up to before 1980, there was not much variation. But after 1980, you can see temperature is increasing very sharply. And the over ocean also and over land also. But here you can see the increase in temperature over land is higher than the increase in temperature over the ocean. So this way we can say it real warming is taking place. So here again I want to show you model result. In this model, we have shown the effect of natural gases and natural activity and anthropogenic. So if you look the picture of the variation of the blue line, so here you can say this is the natural variation. It means that nature itself come equilibrium after some year. So this will not contribute much in the global warming. But if you look the black line and red line, so red one is the anthropogenic activity and black one is the observation. So here you can see in the model, it is showing that anthropogenic activity is more responsible for the global warming and natural activity is not responsible for that. Then why is India concerned about the climate change? So India is concerned because our main work is more than 70% population are living in the village area and they are totally depend upon the agriculture on their livelihood. So if there is a reduce in the production of the crop, then it has direct impact on the economy of the country. Sea level rising, that also leading submerge a highly productive coastal area and many people are also vacant that place. Increase in the frequency of extreme events like cyclone, flood, and drought conditions. So many results have shown that during recent few decades, the frequency of extreme events and cyclone are increasing, and flood and drought conditions are also increasing. This had direct impact. And then finally, it had the water resources is also reducing. It has impact on our concern. So here I want to show the 26th July 2005. That was a very severe rain occurring in just 24 hours. In one day, the 96 centimeter of the rainfall occurred. So this looks the Bombay, that is a very central Bombay area. So you can see the most of the buses are even merged in the water. And this is that taxi stand and very close to the central Mumbai area. So here also you can see. And this is the outside area of the Bombay airport. This is the parking area just behind the Bombay airport. So you can see huge amount of and most of the four-wheeler are immersed in the water. So according to report, the total number of deaths due to this heavy rain was near about 1,000 people died, and 5,000 crores of rupees was estimated to loss in the state. And there is no electricity in the certain area of Mumbai for the last 5 days. And 1 million people rendered homeless, and about 11,000 flights cancelled. And airport was closed for 2 days continuously, and 5 million mobile and landline was crashed. So why the vulnerability increasing? You know, nowadays rising in the population is a big factor. So what causes? People are observed urbanization. They don't care where we are going to build our building. So they don't care about the coastal area. So this activity will create development of high risk zone, and then environmental degradation by the deforestation, and finally it caused the climate change. Then I will come to our research. I want to show you. So first up, I want to show you some observational result. So here I have taken the data from the India Meteorological Department from 1975 to 2007, and I have used 23 stations all over the India, which cover the entire India region. And here I have tried to show the hot days, cold days, hot night, and cold night. What is the effect of the So hard day is defined the days with maximum temperature above 90th percentile, and cold day is defined the days with maximum temperature below 10th percentile, and hot night is defined the night with minimum temperature 90th percentile, and a cold night is defined the night with minimum temperature 10th percentile. Here important thing is that whenever you are going to define the hot days and cold day, so you have to use the maximum temperature data set. And when you are going to define the hot night and cold night, you have to take the minimum temperature data set. So if you look the trend of the maximum temperature, so this is the winter season and this is the pre-monsoon season and post-monsoon season, and this is the monsoon season, post-monsoon season, and annual. So if you look this trend, so you can find that annual mean maximum temperature shows significant increasing trend at the rate of 0.22 degree per decade over all India, and about 39% of the stations are increasing. And if you look in the seasonal wise, so you can find maximum increase in temperature was observed in the winter. And if you see the minimum temperature and trend again from the same season, so you can see that annual minimum temperature shows significant positive trend at the rate of 0.2 degree per decade over India, and about 2/3 of the station show the significant increasing trend at the 5% level. So if so you can for the same season, so you can see that annual number of occurrence of hot days in India is increasing, and its rate is 5.5 days per decade. It means that in 10 years, the number of days of hot day will increase by 2.5 days. Similarly, if you see the trend in the frequency of cold days, so here you can see the cold days is decreasing. So annual frequency of cold days over India is decreasing at 2.4 days per decade. It means that in 10 years, the number of cold days is going to decrease by 2.4 days. And you can see that hot night is going to increase and about 12.9 days in a decade. It means that in every year, the hot day is going to increase by 1 day. So similarly, so cold night is also going to decrease. So 9 days per decade. It means that in every year, almost 1 day is going, cold night is going to decrease. So now I want to come to the experiment, model experiment, what I have done. So I have used the AGCM model, and I run the model at 40 kilometer horizontal resolution. So for the historical period, I have run 1979 to 2008, and then I have run up to 2100 year. What will happen at the end of the 2100 year? I'm not going to give the detail about the model. So just I want to show the result. But for that, when you are running the model, you require huge infrastructure facility. Without high computing power, you cannot run the model. So here I want to show one example. Suppose if you want to run the model for 1 day, you want to project 1 day simulation and run 500 to 600 kilometer resolution. So, and so it will take 1 day. And number of grid point will be 1920. But if you want to run at a very high resolution model, so 30 kilometer by 30 kilometer. So for 1 day simulation, you will require 3000 days. So suppose if you want to run for 100 years, so you require not only one computer, you require series of computers. So this work I have done during my stay in near about 500 computers at a time, and this run was near about more than 4 hours, continuously 24-hour run. So you can imagine how much time takes place to study the climate change. So this is the model result. First, I will show the reliability of the model. So here, this is the observation. Hello. So you can see the ITCZ, Intertropical Convergence Zone. This is the area. Model is also well captured, the ITCZ. Then, and the seasonal variation, latitudinal variation, also you can see the red one, that is the model, our model. So you can see the model is very reliable. So now, with the help of this diagram, I want to show you the how much temperature is going to increase up to 2100 years. So this global warming is taking place not only in India, but it also takes place all over the globe. So at the left side, this is the 4.5 RCP 4.5 scenario. It means that control carbon dioxide emission in the atmosphere. And right side is the 8.5 RCP 8.5 scenario. It means that high concentration of the carbon dioxide in the atmosphere. So, and in this picture, you can see the variation like 2078, 2085, 2091, not only in India, but all over the globe. That is why most of the scientists are very much worried and give the time to time, give also send their result to the government to prepare the proper policy to reduce the impact of global warming. So now this is the annual temperature over India. So you can see the red one, that is the RCP 8.5, and the green one, RCP 4.5. So this way you can see that temperature is rising very tremendously, and up to 2100, the temperature will rise. Temperature will reach near about very close to the 4.5 degree. So you can imagine in India we have recorded the temperature of 45, 46 like that. At that time, situation was very burning type of situation. So now they imagine if temperature will rise 5 degree, means now near about 55 degree, then you can imagine what happened. So same way, we have tried to show the increase in temperature over India in different year. So in different year also you can see the temperature in RCP 4.5, it will give more than 3 degree. And here we can see some of the year have showing the temperature of 5 degree. And this is the annual precipitation. So here you can see the precipitation is not showing the increasing trend, but it shows the decadal change. Like here, you can see increasing, decreasing, increasing, decreasing. Some decade it is increasing, some decade it is decreasing, some decade increasing and decreasing. And here you can see in India in different year how at this time. So here, the what will change in the atmosphere? That is the GMA. So you can see that east and west part of the India in the per day we are getting highest precipitation over the east part of India. But future in the western part of India. So how many percentage of scientists are agree for that? In 1991, only 60% of the scientists, they think that the global warming in was not agree for this. But after 2007, you can see that 97% of the scientists are agree that the global warming is taking place. So here you can say what is the status of the scientists. So what we reduce the vehicle use, better to go by bus, because if everyone go by his car, then the concentration of the hydrocarbon will be increased in the atmosphere. Just hello. So reduce the vehicle use and carbon capture and
Yeah, hello.
yeah.
Hello, Dr. Singh, may I request you to conclude because we have next speaker.
Excuse me, sir.
May I request you to just conclude?
Okay, okay. I am coming to conclusion only. Okay. And recover and decrease in the and tele connection. What I want to show you, if you take the Indian monsoon rainfall and the East Asian monsoon rainfall, and here I have tried to show the Indian monsoon rainfall with the North China, Korea, Japan, and South China. So you can see in this picture follow the Indian increase in the precipitation over South Asia, then there will be increase in the, but this has shown and even southern China also show the inverse relationship. Similarly, you can see in the RCP 8.5 scenario, it shows the very similar trend. So, so I want to come to the conclusion part. So annual mean maximum and the minimum maximum temperature will increase by 0.22 degree per decade, and minimum temperature will be increased by 0.2 degree per decade over India. Occurrence of hot day and cold day is decreasing significantly at the rate of 2.4 days per decade. Hot night over country so decade over the cold night show the decreasing trend at the rate of 9 days per day. And the projected summer precipitation from different time slice experiment, they depict an increase in the northwest India and the west peninsular India, and a reduction over the northeast and north central India. And a correlation pattern of precipitation over South Asia shows in phase relationship with North China and out of phase relationship with Korea, Japan. While the precipitation variation over Korea, Japan, and southern China shows out of phase relationship with the South Asia. So whatever I told, discussed today, you can find all things in my this recent publication here. So thank you for your kind attention.
Thank you, Dr. Singh, for in fact, the reasons behind. I request the audience to limit the question to one or two. there is any.
One minute, sir.
So I think because of your well elaborate talk, satisfied and thank you for your talk. Thank you for.
Thank you, sir. Thank you. So,
No, sir.
and if there is no question, then I can quit now.
नहीं मनोज सर नहीं next one.
Presentation will start. Thank you for listening. You may quit, please.
Okay, okay. If there is no question, then I can quit.
मनोज सर नहीं फोन कर लेते हैं उनको तब तक किसी को बुला
Presentation now.
yes
By Narayan Mishra, Mehra.
नारायण नहीं है next sir.
The second one is by Narendra Sahu. Policy composition and is Narendra there?
no next sir. we'll check sir.
Is Kalpana Mishra there?
yes sir.
May I invite Kalpana to present the talk? Is Kalpana there?
मतलब so next
Next is Lily Rose Beck.
yeah please also there. हाँ है
Lily Rose Beck. Are you available? Presentation. Now, please. Limit with the time, please. What is article not to go there?
Traditional medicinal plants for treatment of body diseases in tribal communities, state of traditional health practices is among the traditional medicinal plants that are used by the tribal community for prevention of they had enough knowledge about the medicinal plants. They look at life, and they follow their own workplace practices and especially using of good patients and I have the material and movement and I have the survey studies and I have selected 5 periods of the report. I have information what they say. I have the local infra local it's unfold it's one of indicators আমরা freelancer data recovery technical experience and
Morning, Namaskar. Dr. Dubey is Joint Director, Indian Plywood Industries Research and Training Institute at Bangalore. At the like and award for some assignment and training institute. He did some may I request? Thank you so much. हो गया सर हो गया फिर से उनको
फिर से वो हम ही लोगों को कॉल करेंगे। तब तक स्टॉप डाल देते हैं, वो डाल देते हैं प्रेजेंटेशन। अरे लेकिन आपकी आवाज वहां से नहीं आ रही है। ये पिन हो रहा है ये वाला ऑन करूं कर यस सर सर के पास है ना। अभी किसको बुलाना है बताओ ना सर को।
Is Nusrat ul Islam available for presentation? Or anyone from her group? Subira Rafiq, Gauhar Ali, Jade Dar, M.A. Rache, Altaf Wani, Rumi Jan, Rehana Akbar. Anyone from this group?
Sir, Pragati Shukla, P. Shukla.
Next. Shukla is the next. So
Yes, sir.
if there is none from the Nusrat group, we can switch over to Parvati Shukla. Pragati Shukla. So
Sir, may I start?
Yeah, Pragati, you can start.
Good morning, everyone. A very good morning to all of you.
Pragati,
I would like to.
I request you for one thing. You limit your talk for 5 minutes. Only, right?
Yes, sir, sure. Yes, sir, sure. A very good morning to all present here. I would like to start my presentation. Respected chairperson of the session, I'm very grateful to the organizing secretary and to the vice chancellor of the university for giving me an opportunity to present my and also to the convener. My topic is I'm a research scholar at Hemchand Yadav University, and I'm working at the research center St. Thomas College. Under my guide is Dr. V. Shanti, head of department of microbiology. My topic for today's paper presentation is collection and identification of the wood decaying fungi, that is polypores of Bhilai area. These are my contents: abstract, introduction, review of literature, material and methods, results and discussion, and my references. Now, fungi, as we know, they are the second largest kingdom, and basically, about the fungi I'm talking about are wood decaying fungi, and they are commonly associated with fallen woods, and they infect trees, and they are responsible for wood decay and wood rot. They basically belong to two phylums, Basidiomycota and Ascomycota. They are basically tubes or pores bearing, and they are excellent nutrient recyclers. My study was to collect and identify these wood decaying fungi from Bhilai area, and the main families that I have got are Polyporaceae, Ganodermataceae, and Hymenopteraceae. And the main fungi that I have identified are Felinus species, Lenzites species, and Termite species. Next are, as we all know, as we are in the symposium based on biodiversity. Biodiversity is a term which is referred to the genetic, taxonomic, and the biogeographic variability within a species, within a specific area or within a region as a whole. And since these particular wood decaying fungi have not been studied much in central India and especially much in Bhilai, hence I have taken this topic. The wood decaying fungi, they are of two types, white rot and brown rot, and they are excellent carbon recyclers, and they are also being used as mycomedicinals. That is, they are having these antimicrobial compounds which are responsible for antimicrobial activities, and many antibiotics have also been developed on through them. Now, if we see the review of literature, there are many scientists who have been working on the biodiversity. Recently, two I have been able to find out, that is Adarsh et al. And Darwana et al. Both have studied polypore species and have identified various genera, and they, like Adarsh et al. 2019, he has found that white rot species are dominant over brown rot, and he has found 57 among 57 species, 42 were white rotters. Similarly, Darwana et al. Has also worked, has also identified fruiting bodies, and he found that he was able to identify 71% of the polypores from the total collection. My material and methods, I had started collection from July to October, since they are found only on in these between these months, and I have screened fallen woods, I have screened trees, and then I have collected the fungi. Then I have they are being brought to the lab, cleaned and sun dried, and they have been identified on basis of macroscopic characteristics, which has been given in the key by Bakshi et al. Now, results and discussion, a total of 66 specimens were collected, out of which 54 were identified, as some of the specimens were damaged, and some were already infected by secondary pathogens, so they were not identified. Basically, all the fungi belong to the order Polyporase, which comprises of five family, that is Polyporaceae, Ganodermataceae, Hymenopteraceae, Serpulaceae, and Fomitopsidaceae. Now, the if I see the major genus or the major species, they are Daldenia concentrica, Ganoderma lucidum, Xylaria polymorpha, Felinus badius, Felinus gilvus, Polyporus versicolus, Polyporus ramocephalus, and Scythophilum commune, and Termites singulata, Termites cubensis. Among these, the Felinus was the most dominating one. That is, there are 5 species of Felinus which I have collected. They are Felinus badius, Felinus gilvus, Felinus alardi, Felinus pachyfloeus, Felinus ignarius. Then, since as you can see that in my study, there are a variety of polypore fungi which I have collected. But if we compare with other studies, there have been studies which have been solely focusing on a single polypore, like Shirsagar et al. 2009 and Bhosle et al. 2010. They have focused solely on a single polypore, while in my study, we have got a diverse variety of polypores. These are the locations from where I have collected in Bhilai, and also I have done in Durg, but this paper solely focuses on Bhilai. These are the on location snaps which I have clipped while collection. While collecting, I had taken these, since they are attached to wood logs or they are found on trees, so I have taken the snaps, and the rest of them are the snaps which I have taken from the somehow this is not moving. Hello, are the slides moving? Hello. Yeah, these are the snaps of the These are the snaps of the connected polypore. And lastly, my conclusion. From the 66 samples of polypores, 54 were identified. And as I told you, the major genera were there, but still there is a need for extensive study on the polypore biodiversity. These are my references. And I would like to acknowledge three persons, that is Dr. C.K. Tiwari, who is the senior scientist from Tropical Forest Research Institute, Dabalpur, who has helped me in identification. My guide, Dr. V. Shanti, Head of Department of Microbiology, St. Thomas College, for all the inspiration and guidance. And my sincere thanks to Dr. M. G. Royman, Principal, St. Thomas College, Bhilai. I would also like to thank the organizing committee, the convener, and of this symposium. Thank you so much for giving me an opportunity. Thank you.
Thank you
Good day,
very much
everyone.
for your presentation. Just a brief query. You have
Yes, sir.
you have collected a number of species in Bengali. Have you cultured them or you have identified?
Sir, actually, my topic, my PhD topic is antimicrobial activity. So I haven't gone to the mycelial level. I have just collected. And then I have cleaned and extracted. I have gone for extraction directly without going to the mycelial level. Because sir, my topic is antimicrobial activity, so I am focusing on that.
So this is just a suggestion. If you have to the antimicrobial activity, they have to define the culture media required to grow these.
Sir, I'm not able to get your question. The voice is, I don't know, it's echoing. I'm not able to understand the question.
Question was,
Hello?
if you have collected all these in Bengali,
Ji, sir. Yes, sir.
They are known as
Sir.
lignicolous.
Lignicolous. Yes, sir.
And if you want to look at the antimicrobial activities, then you have
Yes,
to
sir.
culture them in the laboratory.
Sir, actually, I have actually powdered the fruiting bodies, dried and powdered.
Okay. That means directly extracting. Okay, thank you. Thank you very
Yes,
much.
sir.
Thank you.
Yes, sir. Thank you, sir. Good day.
So, I think another people will go back here. And, am I audible you?
Yeah, you're audible just, but voice is not very clear, but I can hear.
Okay.
And how about my voice?
Yeah. So I think Dr. Dhuria has already introduced Dr. Dubey, and he was one of our past colleagues in, I think, early 2009-10 or so. So I request Dr. Dubey to present his talk.
Okay.
And your voice is now audible, so we can go ahead. Please.
Okay, that's good, sir. Thank you, sir. Let me grab my presentation. Are you able to see the slide? Are you able to
Yes, sir.
see? Okay. Okay. So thank you once again, and again I like to thank Chair Dr. B.N. Tiwari, Co-Chair K.K. Chandra, and all the faculty staff of Department of Forestry, GGB, and faculty of other department, as well as dear students. Before I start my talk, I just want to briefly introduce my institute, that is Indian Plywood Industry Research and Training Institute. So we are an autonomous body under Ministry of Environment, Forest and Climate Change, Government of India, with a 3 centers in Bangalore, Mohali, and Kolkata. And basically, our role is to promote panel industry development. That is the development of plywood, particle board, and MDF industry, to promote export of domestic panel products, Also to conserve the forest, because we are utilizing wood or raw material which is growing outside forest, so we are contributing in that aspect also. And then we are also involved in the promotion of MSME. This all plywood and panel industry, they comes under MSME. So we are also support the policy towards the development of this industry. We also give skill development training to meet the human resource requirement of the industry. Then we also develop BIS standards for different wood products. We promote wood as a green building material, and also other non-conventional materials such as bamboo and agro residues. So today I am going to focus my talk on what is the role of renewable material which is coming outside forest in biodiversity conservation through reducing pressure from forest, as well as providing raw material to the industry. So we can see that human live or went into the forest and look for the best suited tree or part of tree which satisfy their basic need of housing or burning or cooking, hunting, shipbuilding, or for social and religious purpose. And with the time, people learn to modify or improve some of the undesirable feature of wood, because wood also shrink and swell. It is also attacked by fungus and insects. So through this technology, we can overcome some of these shortcomings, such as seasoning, chemical treatment, application of coatings, sawing, as well as veneering. However, during last 40 years, the wood utilization scenario is changing in the world as well as in our country. As you see, in the 1980s, the more focus was to replace wood with the other substitute like plastic, aluminium, steel, or concrete. So that was the theme during 80s, and heavily propagated by plastic lobbies that better to use non-wood products. However, as witnessed by the previous presentation, that it's resulted in significant increase in the pollution from plastic, as well as waste material from concrete or steel, and it resulted in the loss of biodiversity, as well as global warming and other impact to our climate.
Okay, the slide is not moving.
Okay, so we are in which slide?
First slide, we are still stuck at first slide.
Oh. Let me try again. Can you see the slide now?
No, it is third slide. Third slide.
Okay. Now, can you see the slide?
Yeah, it is.
Okay. We were talking that the human use wood for various purposes since ages, and then to overcome some of the undesirable feature of wood, we developed technologies such as wood seasoning, chemical treatment, application of coating or paints, or sawing or veneering. However, scenario regarding wood utilization is changing during last 4 decades. In 80s, the focus was more on using a substitute of wood, such as plastic, aluminium, steel, or concrete, because this was the theme that replaced wood products from non-wood products. However, it resulted in significant pollution of our sea resources and land resources, loss of biodiversity, and impact in the form of global warming, which we all are talking about these days. However, the perception of wood, utilization of wood is changing gradually, because now wood which is growing outside forest, that is very important. It is considered good for environment and society, because wood can produce by using sunlight, water, and carbon dioxide with a minimum energy. We can produce the wood while for steel and concrete, it requires a lot of energy to produce steel or concrete material. Through the latest technology, we can utilize now each and every part of wood to make various value-added products. So there is no wastages, and wood can process by simple tools, by handheld tool, or with minimum energy. We can operate those tools as compared to steel and other material. Growing wood along with agroforestry is also contributing to for the additional income of the farmer. It can be a best alternative for the traditional wood coming from the forest. Wood is biodegradable. That is no issue of degradation after the end of its life cycle. Wood also synthesizes carbon and lock the carbon in the form of wood products for 20 to 30 years. So it is helping in carbon sequestration. Then wood construction is very good insulation material. Like in cold countries, most of the houses and construction is of wood, so it saves the energy. Otherwise, if we are building our house by concrete, we have to always heat the home the temperature minus 20 or minus 30 degrees centigrade. So it saves lot of energy. So in the nutshell, now wood is considered is very good for the environment, and the thrust should be grow more wood and use more wood. And during last couple of decades, with the growth of our population, growth of our economy, rapid urbanization, and reduced innovation cycle. Like at the time of my father or my grandfather, we used to get our bed or furniture for 30 to 40 years and transfer one generation to another. But now this new generation, they want to change their furniture in 8 to 10 years. So all this resulted in the increase in the demand of wood and wood products from 58 million meter cube to 153 million meter cube by 2020. And this round wood demand for industrial is about application is about 57.2 million cubic meter, 12.5 from paper industry, 11.8 from plywood industry, 24.4 million meter cube from construction industry, and 8.5 million meter cube from furniture industry. And by 2030 also, it is projected that the timber demand will increase by 98 million meter cube because of the government thrust for housing unit, and government of India is aiming to provide housing for all Indians. And also there is a prediction of growth in furniture sector. So this all are resulting in increase in the demand of wood and wood products. But how much wood we are getting from our forest? So our forest area is about 23.3% of India's area, and supplying only 3 million cubic meter of wood, while tree outside forest now it is supplying significantly chunk, about 85 million cubic meter.
The slide is again stuck up at slide number 5.
Okay, the slide is not moving.
No, sir.
So which slide you can see?
Yeah, 5. Slide number 5. Now it's 13.
So now you are saying 1980 slide, or which slide?
It is 7th slide visible. आपने slide show किया है क्या? आप देख नहीं पाएंगे, मुझे लगता है slide show में.
Okay, I'm not going for slide show now. Okay.
Actually, yes.
So now you can see this source of wood produce.
Yes, yes.
Okay.
14.
So from forest we are getting about 3 million cubic meter of wood, while from tree outside forest it is 85 million cubic meter. And this table gives you the overall demand and supply scenario. Demand of the wood products is increased from 50 to 153. Output from forest, it drastically reduced from 24 to 3 million cubic meter only, while output from plantation is continuously increasing from 41 million cubic meter in 1996 to 85.7 cubic meter by 2020. And overall, there is a deficit of 64.3 million cubic meter. So how we can meet this demand and supply gap? One alternative is we can go to import the wood. That is importing from other countries, and currently we are importing about 6 million cubic meter of round wood and sound wood products, 15 million cubic meter of wood and wood products, including furniture. That is costing 44,000 crore rupees to our economy. So this import of wood is meeting about 5% of our wood requirement, which is increasing, which is also increasing our import bill. And the export countries like Malaysia, Indonesia, they are also imposing ban or restriction to the export from their country. And this policy of import is also not in line with Atmanirbhar Bharat or Make in India initiative of Government of India. So next alternative, which is a more viable alternative, is to go for renewable material outside forest, which mainly include tree outside forest, bamboo, and agro residues. Like at Ipati, we have utilized the tree outside forest like eucalyptus, poplar, Melia dubia, silver oak to make a range of wood panel products such as plywood, particle board, plus door, blockboard, etc. And we have also contributed to develop industrial hub based on eucalyptus and poplar in Yamunanagar in Punjab, Haryana, and Uttarakhand area in partnership with farmers and industry. And another hub we have developed in southern India using Melia dubia in Karnataka or rubber wood in Kerala to use this fast-growing species to make wood panel products. Our country also has significant bamboo resources, but as compared to China, our market is very less. Like the market for bamboo product for China is 30 billion US dollar, while our is only 5 billion US dollar. So there is so much gap, and we have developed range of products. Not only developed products, but we have also patented those products and transferred to the industry. So bamboo can be also one of the resources which can meet the raw material requirement of our industry. And apart from this product, we have also developed various type of bamboo housing, which can also contributing in the Prime Minister housing scheme, as well as for disaster-prone area. Bamboo housing can be a very good solution. So this all our houses are developed at Ipati. Apart from that, various type of agro and forest residues such as rice husk, rice straw, wheat straw, bagasse can be developed into various type of panel products, as well as forest residues such as pine needles and lantana can be also converted into various type of panel products. Recently, we got patent on this rice husk particle board, and which can be a viable solution for air pollution in northern India because of the burning of parali. However, there are certain issues with utilizing this raw material from outside forest. If you see the raw material from forest, they are very large in diameter and their quality is very high. As compared to fast-growing species, their diameter is very low and they have very minimum amount of hardwood. So their properties and qualities are very inferior compared to the forest-grown wood. And similarly, there is a technological challenges to use sympodal bamboo. Like China bamboo is monopodal bamboo. It doesn't grow in cluster. It grows separately. While our bamboo is sympodal, so it requires more processing and more input of technology. Similarly, agro residues and forest residues also need specific technical input. So the first is that this raw material is low in quality, but with the intervention of technology through the Institute of Ipati or FRI or IWST, we can overcome these shortcomings. There are some leverage opportunities also for utilizing this raw material from outside forest. First, this all material is based on renewable or recycled material, and it can contribute to the closed carbon cycle, as well as it can contribute to towards the achievement of goal of bio-based circular economy. And there is a predicted growth in the sector due to predicted growth in the furniture and housing sector in the country. And industry has also diversified raw material base from forest grown wood to tree outside forest. Now industry is also accepting this new raw material. And there is already proven agroforestry model established by various paper and pulp industry, which can be also emulated by other industry. There is an institute exists like ICFR and IPRT, which can provide research and training to utilize these resources. And Niti Aayog, it's also predicted that bamboo resources in our country has a potential to generate economic activity of more than 50,000 crore and subsistence employment opportunity also. And recently, Ministry of Environment Forest also declared bamboo which is growing outside forest is no longer considered as a timber. Technically, bamboo is a wooden grass, but Indian Forest Act consider it as a timber. But now, bamboo which is growing outside forest is considered as a timber. So our farmers can grow and without any hassle of cutting or require any permit for cutting the same. And similarly, some startup and all also showing, especially in the Bangalore, we are getting many startup which are showing interest in using this non-conventional material. There are some challenges also, like the raw material supply is very inconsistent, the quality of raw material and inefficient utilization. The industry, panel industry is not very organized. It is highly fragmented and unorganized industry. Day by day, the environmental concern are increasing. The policies regarding forest is changing, and these are creating lots of problem for the industry. Likewise, the requirement of certification from FSC or other organization. And there is one more thing: there is a perception of society towards wood and wood-based industry. Like still, many officials, many eminent people in society, they consider wood as a not good for the environment, and wood-based industry has a negative image which need to be changed. And there is a global competition from cheap import, and the skilled human resource is also limited, and there is an inadequate infrastructure and human resource in the country.
Dr. Dubey, may I request you to please conclude?
Yes, sir, only 2 slides, sir. So we require certain policy intervention, such as encourage production of wood growing outside forests, to give some incentive to the industry in the form of some tax benefit, and treat wood form as a agriculture farming. Like it should be wood which is growing in farm. It should be declared as a agriculture product, not as a forest product. And then research and development required in treat outside forests. So finally, it can result in increase in productivity of land and resources. It can increase employment, increase income to the farmers, reduction in rural distress. There may be more saving and earning foreign exchange, and increase taxes to the government if we promote this raw material outside forest. And it can also contribute to achieve the 33% target of forest and tree cover in the country. And finally, all this will also going to contribute to conservation of environment and biodiversity, because we are not touching the forest wood, we are just focusing outside forest, and also promoting the wood and wood products. So with this, I like to thank you for your attention and giving me the time. Thank you. Any question?
Dr. Manoj, thank you very much.
Hello? I'm not getting your voice.
Dr. Manoj,
Yes.
thank you very much for a very nice presentation.
Thank you very much, Professor Singh, sir.
Was here for 2.5 years. Then he has gone to USA my very prestigious fellowship. He was there for 4 years. And I tried my best that Dr. Manoj to come back again and join this department. But he has not joined this department. And you will be surprised to know that he has been selected assistant professor in IIT Bhopal in Australia. And recently he is joined the research in the Institute of Plywood and Training Research in Bangalore. He is a person who has done a PhD from FRI Dehradun, then PhD from Cambridge, UK, again, interesting PhD from Germany, in all forestry systems. So he is a quality person in the subject, and we can say we are proud that he is our country here. And still I am trying that how Dr. Manoj will join this department. We hope Dr. Manoj, you will be rich in the future also to us.
Yeah, thank you very much, sir. Though I'm not able to hear you clearly, but I can understand the essence what you are saying.
Are you listening me? Are you listening me?
Yeah, not very clearly, sir.
हेलो हेलो आवाज नहीं जा रही है क्या?
हाँ सर, आ रही है पर बहुत साफ नहीं आ रही है. I can get your essence what you are saying, sir.
ठीक है बहुत अच्छा लगा. और कोई सवाल पूछना चाहे आप?
Sir, I have a question.
Yes, Anurag, please.
Okay, thank you.
Good afternoon, sir.
Yes.
Sir, as you know, in Northeast India, especially in Arunachal Pradesh, the natural bamboo forest is there, and people are using those natural bamboo making their houses and other crafts. Sir, my question is how they can use those bamboos for their livelihood income generation, because they are using for their own purpose only, but till now they means there are very less use for their livelihood income generation. So how means in broad sense how they can use it for their livelihood income generation?
Yeah, it is very pertinent question, Anurag, because as you rightly said that Northeast India, there we have maximum more than 70% resources of bamboo are over there. But for livelihood generation, so far we are only depending on the making some furniture item or some small handicraft. We have to think about the industrialization of bamboo. Like we have to think about the making the bamboo wood or bamboo based higher value industry. So this local community can supply the primary material for this industry. Like in Kerala, many these tribals and women, they are supplying mat material to one of the bamboo based industry, and they are getting very good payment from the forest department. And this Kerala Bamboo Board, they are making bamboo product from those mat and then supplying to the market. So we have to think about the one model where the community can be involved to produce a primary material as a raw material for a subsequent industry. So in that way, we are also trying that we have established 7 to 8 primary processing industry there. So there we can involve community also.
Okay, thank you, sir.
Any further question?
बस 5 मिनट बाहर गया उड़ गया ये वाला ये
So if there is no question, then I like to thank especially Professor S.S. Singh for giving me this opportunity and always showing his love and concern, appreciation for me, and through his support, not only his and but other faculty of Department of Forestry in GGV, I contributed in this field. So again, I thank to the organizing committee, especially Professor Singh and Dhuria sir, Tiwari sir, Chandra sir, for inviting me. And I wish all the success of the rest of the workshop. Thank you.
इनपुट निकालो ये माइक ये सर ये लगा ये वाला उसमें वो वाला ठीक है इसको लगा लो ठीक है ओके कनेक्ट वो निकाल तब तक अपने
Hello? Hello?
Hello, please. There's a technical issue. Please give us 5 minutes. Hello. 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 Am I audible?
Yes, sir.
Yes, sir.
Yes. There's a technical issue over here, so kindly give us 5 minutes.
Sure, sir. Thank you.
Hello. Hello. Yeah.
हेलो 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 इसमें आवाज आ रही हेलो हाँ माइक वायर वाला चाहिए वायर वाला यस कर रहे हैं सर एक वायर कनेक्ट कर मेरी सब गिर गई
So the speaker, Gopi Pulalia, are you ready? So just within 5 minutes, we will start to lecture. After Gopi Pulalia, then Kruti will be also ready. After the Gopi lecture, you will also give your
Hello?
Hello. Sir,
प्रेजेंट कर रही है उसको मना किया गया है तो रिमूव कर दिया
my, hello. Hello.
hello. Gopi Pulalia is ready.
सर वहां जाइए उनको वो बोलिए नेक्स्ट सब्जेक्ट ठीक है वेट
आवाज नहीं आ रही Gopi Pulalia.
हाँ चैट में टाइप करें गोपी को लगे नॉट ऑर्डिनरी हाँ नॉट ऑर्डिनरी
problem will be the brain technical. ऑडियो नहीं आ रहा है, ऑडियो नहीं आ रहा है, आवाज नहीं आ रही है।
Hello?
Yes, हेलो, मेरी आवाज आ रही है?
Hello?
हाँ, आप बोलिए, आपकी आवाज आ रही है।
Sir, it was my third turn, but you are not allowing me to join, sir. Hello?
मेरी आवाज सुन रहे हो आप? मेरी आवाज आ रही है? उनकी तरफ से टेक्निकल प्रॉब्लम है, यहाँ से कोई टेक्निकल प्रॉब्लम। कल भी यही प्रॉब्लम हो रहा था उनके साथ। अभी तो ये इसकी आवाज आ रही है, यस सर, नुसरत इस्लाम की आवाज आ रही है। हाँ, दूसरा गजन वो रेडी है, उसी को बोल बना जो बार बार टाइप कर रहा है। गजन को बोल दिए। हाँ, सर आप बोल सकते हैं, आपकी आवाज आ रही है। आवाज आ रही है आपकी ऑनलाइन, यहाँ से साउंड नहीं आ रहा, तो हम जल्दी करने की माइक वाला वायर लाने एक केबल लगाना चाहिए।
Mr. Dr. Gajendra, are you ready for presentation? Alka Gupta present.
Yes, sir, I'm here. Yes, sir. Yes, sir, I am here. Can I present my presentation right now? Okay. दीपू प्लीज उनको मना करो. हेलो. हेलो. Yes, sir. Yes, sir. My slides are visible. Okay, sir. Okay. Very good morning to all of you. Hello?
Can you be a little louder, please?
Yes, yes. Very good morning to all of you. I am very thankful to get the opportunity of presenting my paper here. I am Alka Gupta, research scholar from the Department of Botany, Banaras Hindu University, and working under the supervision of Professor R. Sagar. Today, I am going to present a paper on the topic entitled to establish regression equation between herbaceous cover and biomass in dry tropical grassland for non-destructive estimation of biomass. So, the first question is why this topic was performed, why this study was performed. Generally, biomass is harvested by harvest method, which is destructive and could lead to severe loss of biodiversity. And as we all know, every plant species has its own significance in an ecosystem, and loss of biodiversity could cause detrimental effect on balance in ecosystem sustenance. And for assessment of biodiversity loss, biomass and cover are important measures, but still there is lack of such studies from tropical grassland. So there is a need of indirect method of biomass estimation, and if we search of indirect method, then plotting regression equation could solve our problem. Just by calculating the cover, we could get the biomass of that species by using regression equation. So what is biomass? Biomass is total dry weight of plant present in an area, and cover could be simply defined as how much aerial part of the plant is covering in the quadrant. Let's come to the objective. There are two major objectives of this study. First one is to collect herbaceous plant sample, estimation of its biomass and cover. And second one is to draw species-wise regression equation between plant biomass and cover. This is my study site. This slide shows the study site, which is located in campus of Banaras Hindu University at 25.31 degree north latitude and 82.97 degree east longitude, and 80.71 meter above sea level. Samples were taken from the whole campus of BHU. Methods and materials. Samples were collected from the campus during November 2019 to February 2020. For calculation of cover, 1 into 1 meter square quadrant was taken, and it was further divided into 100 cells, each representing 1% cover. Biomass of each species was measured by harvest method. Plants of 1 into 1 meter square quadrant were harvested, oven dried, and weighed. These data were used to establish regression equation with the help of appropriate statistical procedure. Result shows that 119 quadrants were sampled and 69 different species were found, belonging to 28 different families. Asteraceae family has highest species number. Relationship between biomass and cover is mostly linear, but it's not always same. In case of, as in the case of Dicanthium sida ageratum malvestrum, equation is linear, but in the case of Zephyranthes, Ruellia, Oplismenus, Iragrastis, Abutilon, Nephalium, Stoncus, Vernonia, and Nicotiana, power regression equations were found. But in case of Uraria, Andrographis, Chenopodium album, they showed exponential regression equation. And if we talk about R square value, it varied from 0.38 in case of polynomial equation to 0.99 in case of linear equation. In the power equation, linear equation, polynomial and exponential equation, the maximum R square values are 0.95, 0.99, 1, 0.93 respectively. And maximum R square values are 0.42, 0.50, 0.38, 0.056 respectively. This slide is showing the graph between biomass and cover of various selected dominant species of PHU campus. Although total 69 graphs were made of 69 different species, but it was not feasible to plot each of them here. So I have selected 16 dominant species and have shown their graph here. Here on the x-axis, cover is shown, and on the y-axis, biomass is plotted.
Alpha Gupta, are you listening me?
Yes, sir.
Your time is over. Please conclude within a minute.
Okay, sir. Okay. Sir, just I will take 1 minute. R square values are also written here in case of each species.
Within a minute, you say what is your important findings. What are you wanting?
Okay, sir.
What are decision and details? What is your important findings? 1 or 2, please tell.
Sir, just give me 1 minute. I'm facing some technical issues. Just give me 1 minute.
We are listening you, please speak.
Okay.
Your sound is good, you speak, please.
Sir, just give me 1 minute. What is happening here? Could you tell me?
Don't bother. Your sound is coming, you speak.
Yes, sir.
Your sound is coming, you speak, please.
यार मुझे नहीं आ रहा क्या बोल रहा है
Okay, your time is over, madam. Please, we are going to keep your system off.
What is it? कौन
Gopi Koladia, are you connected? Can you please unmute yourself? You are not audible.
Yes. Sir, I'm not able to get what you are saying.
No, your time is over. We are moved to the next participant.
Okay.
Gopi Koladia, please switch on your mic.
Hello, am I audible, sir?
Yes, you are audible. Please start your presentation. Gopi Koladia,
Am I audible, sir?
Yes, you are audible. We can start now. 5 minutes.
Okay, sir. Now, good afternoon to all. I present my presentation based on diversification of the antibiotic susceptibility and enzymatic profiling of the marine actinomycetes of 2 seasons based on monsoon and post-monsoon seasons from the along coast, Gujarat. Now, I'm moving to the next, mainly the results only. Okay. So now my results is based on the enzymatic potentiality and the enzymatic susceptibility profiling of the most of the 2 seasons, monsoon and post-monsoon seasons. So we can see here, this slide represents the different morphological characters like aerial and substrate mycelia with different medium requirement of the specific isolates. So in this next slide, in this slide show the secretion of the protease enzyme, mostly the extremozymes, protease enzyme followed by the amylase and lipase in decreasing order. So mainly the protease enzyme is the most secretion enzyme of this specific to seasons, monsoon and post-monsoon seasons. So now in next slide, in this slide represent the secretion of extracellular enzyme of two seasons, mainly monsoon and post-monsoon seasons. So the protease and amylase up to the pH 9 and sold up to 5%. Maximum protease enzyme secretion found in this monsoon season seasons followed by the post-monsoon seasons. Now, in this slide illustrated the maximum production of alkaline protease enzyme followed on the post-monsoon season, followed by monsoon seasons. So we can see here in this graph the maximum enzymatic efficiency or potentiality we can shown in this graph. Now, in this slide, represent the data based on a zone of inhibition against 12 different antibiotics of the 13 different monsoon isolates. So we can see here the sensitivity of the whole antibiotic susceptibility profiling against monsoon season isolates. Now, in this slide, we can see here the proof which I mentioned, the antibiotic susceptibility against the whole monsoon seasons. Now, we can see shown earlier. So same as like the post-monsoon seasons. So we can see the maximum or the inhibition against the 12 different antibiotics of 11 different post-monsoon isolates. So we can see here the whole pictures regarding the antibiotic inhibiting profiling here we can see. Now, in this slide, the graph represents the multiple antibiotic resistance index combinedly to post-monsoon and post-monsoon or the monsoon seasons. So the majority of the isolate of post-monsoon seasons resistance against whole 12 antibiotic profiling. So now, based on this data, we can conclude the post-monsoon seasons have a maximum resistance as compared to the most monsoon seasons. But in post-monsoon season, S1 and S2, these two isolates have a sensitivity against whole antibiotic, 12 antibiotic. So now this is my whole two presentation, and now I acknowledge to Professor S.P. Singh sir for his guidance and continuous encouragement. I also thank Ms. Mazbin Sheikh for her active role in this work. Now, financially, also thank to UGC BSR project to support it financially to me as a project fellow project assistant, UGC BSR project assistant. Now, also thank to UGC whole facility and infrastructure under whole project and other programs like UGC CSS, UGC DSR faculty project, DBT, MOS project, DST FIST, DST Young Scientist, and DST Women Scientist also. Now, also thank to Department of Bioscience and my whole SPS lab on my colleagues. So also thank to all to support it to me. Now, this is my whole references, and thank you very much.
Are you listening me properly? Okay, मेरी आवाज सुन रही है?
Yes, sir. Yes, sir. Yes, sir. Okay, sir.
Good afternoon to all the experts. I am Kruti Dangar, and today my topic is that diversity of the non-cultivable proteobacteria of the saline habitat of the coastal region of the Gujarat. And my mentor is Dr. S.P. Singh. Looking to that, a general introduction that saline habitat of the coastal Gujarat. As we all know that Gujarat is located at the western part of the India, and these coastal land cover that 16,000 kilometer area. Lots of industrial activities are also going on in this particular the area. This saline area is highly rich in the microbial diversity as well as the salinity. But looking to the diversity perspective, these areas are just still unexplored. Now, another main aspect is to study in the case of the non-cultivable proteobacteria. So microorganisms, those have the ability or those we have the knowledge to cultivate in the laboratory conditions, then we have to classify it in case of that cultivable microbes. But some of the only that 1 to 3% of the microbes are we are able to cultivate in the standard laboratory conditions. Rest of the microorganisms we have to detect based on their gene from that natural environments. And the diversity we have to judge by with that help of that various bioinformatics tools to identify that what type of the microorganisms are the presence and what they are. Objective of this work is to that the main challenging part to work with that a saline soil is to isolate that a total environmental metagenomic DNA to explore the diversity and the novel proteobacteria from the saline habitat based on that sequencing approach, and the bioinformatics various tool to describe that microbial communities and to quantify that what kind of that communications or linkages are there. Uh, Now we have to go for that materials in the methods, and these materials in the methods at first the isolation of that metagenomic DNA with that help of a mole biopowered soil DNA kit. Then I have to store that I had to store this DNA in the 20 degrees and went for that metagenomic sequencing. In case of that metagenomic sequencing, my aim to identify that what are types of the microorganisms are the presence over there. So I use that only that 16S rDNA targeted gene. I have used this forward and that reverse primer for the amplifications of that 16S rRNA DNA gene to target for the amplification. These were it has been used as the adapter. That sequencing it has been that carried out in that aluminium mic sequencer machines, and then have to identify that all the sequencings I have used these GitHub labs and the MGRAS various bioinformatics tool to capture that what are the microorganisms are the present over there. So looking to that results, here that Devbhoomi Dwarka located in that coastal regions of the Gujarat. Once we are go for that sequencing that total number of that sequences is 22 MB, but we have to that go for that post filter to remove that unwanted to remove that adapter sequencing. So the final reads are that 44,286. After these submissions of that all the sequences, these sequences has to be converted to that operational taxonomical unit. These operational taxonomical unit that will be represented all the microorganisms at diversity, so which is known as that OTU, is for that 2,775. Once we are going to that capture all we are going to put all these metagenomic sequencing into that MGRAS and get it any of that bioinformatics tools, they were giving the different types of the result. The diversity as much as been indicated as the numbers various color it has been indicated over here, and these bar graph result have been shown that in my sample that the proteobacteria diversity is 22%. These 22% means that the relative abundance of these phylum in a particular of the all the metagenomics. After that, diversity in case of that proteobacteria that will be further classified into that alpha proteobacteria, delta proteobacteria, and the gamma proteobacteria. So these, after all these relative abundance should be remain at 22 in case of their class level also. Then we have to, as with the microbiology journey in the classifications of this microorganism from that phylum to a species, that we have to move towards that. So, looking to that order sequencing analysis, the dominance in case of the bacteria class is a bacteria and the order is a Proteobacterius is a 5.6 and 0.1 relative abundance I have mentioned over here. Again, we have to go for that after this order, like the which family it's the most dominant. So in case of these my samples that will be collected from the saline soil of the Dwarka, that family is a Rhodobacteriaceae, it's considering the higher abundance over here, so that we can see that how these further the diversity and the classifications we can easily achieve with the help of metagenomics aspect. Next one, then we have to go for the genus level classifications, and then higher that most abundance that we have observed in my data in the Delta Proteobacteria order is GMID 10, that is also novel, and the species and the family and the genus level it is a 3.3%. Again, we have to go for that genus level classifications, and so every time this classification it has been become a broader and broader, and again that family of the Rhodobacteria it will be considered with the 5.4% of the relative abundance, and the 0.1% it is a Rhodobacteriaceae. So all these things that it is with the higher abundance that has been mentioned highlighted with that green color, and the lower abundance that it will be highlighted with in a 0.1%. So again, this one is the classifications of the genus bacteria. We are not able to capture that what are the species over there because these are relative abundance of these entire Proteobacteria it's a 22%. As for the G sir.
Your time is already over. You please
Yes sir.
conclude.
हाँ sir, last slide are there. Okay.
No, those slides conclude what you can find. Yes.
Okay, sir, that based on that metagenomic approach by targeting that 16S gene sequencing and by using that make sequencer, we are easy to detect the diversity of the Proteobacteria, and not only the Proteobacteria, entire that microbiology community we can also be detected. So this one is a conclusion of this my seminar. Yes, sir.
Your time is over. Now your depends open for the question from audience. Or those who are online, they can ask any question.
Okay, okay, sir.
Okay. I want to know, have you used any marker to identify diversity or any specific pattern of the bacteria, you know?
Also, for that marker, I have used this 16S because it is highly conserved region, and my aim to identify only that bacterial diversity. So once I have to go for that entire metagenomics, what are the just identify what are the microorganisms, but what are the genes responsible for that salinities and what are the regulations? I have to go for that all the metagenomics sequence of that.
Marker to identify the polymorphism like that.
Sir, marker, I use this 16S gene sequence. 16S gene is a marker. Because aim to explore only that bacterial diversity, that's why. If I have to go for that fungi, so I have to use that 18S gene markers.
So are you not thinking to know in a specific diversity, any single bacterial genus or specific diversity in that area?
Sir, actually, once we have to study for that any of that specific diversity, so we can go for that entire diversity result has came. All that bar show the diversity of all the phylums and genus, but I just focused on that Proteobacteria.
Okay, thank you very much for giving me.
Okay. Thank you, sir.
Next speaker is Bhavtosh. Bhavtosh, are you ready?
Yes, sir.
Although have you joined?
Hello. Yes, sir. Yes, I am present, sir.
Okay, so you start your presentation.
Sure, sir. Thank you, sir. Just 1 minute, sir. Is it like my screen is visible, sir? Hello. Presenting this, yeah. Is it visible, sir? Hello.
Not visible, sir.
Not visible. Okay. Once again, sir.
Your screen is visible, but your presentation
If
is
you
not
permit,
visible.
yeah. Yeah, but like I was just okay. Thank you, Anurag. Thank you. Just 1 minute. Sorry, I just stopped sharing. Just 1 second. I think I'll try to go by slideshow. If there is some problem, I'll stop this slide sharing. So good afternoon, everyone. I'm Bautosh Kikani. I hope I'm audible to everyone, and my slides are visible. Hello. Hello.
Yes,
Yes. Thank you.
yes, your voice is, yes, your voice is audible.
So good afternoon, everyone. I'm Bautosh Kikani, and am I not audible?
Yeah, you are audible.
Hello.
Please carry on. Please carry on.
Thank you, sir. Sorry for the like some network issue from my end as well. Good afternoon, everyone. I'm Bautosh Kikani. I'm completed my PhD under the guidance of Professor S.P. Singh from Department of Biosciences University, and currently I'm today I will be director of Symposium. So first of all, I would like to thank the organizers as well for giving me such a fantastic opportunity. This is a very well-known, you can say the research site. It is situated in the middle of the Gir forest, and it has its own ancient value as well. It is well known for the presence of natural hot springs. So we collected water and so chemical analysis of the samples, including pH, temperature, TDS. In case of soils, definitely even the gravels. As far as the glimpse of my in both molecular diversity as well as the biocatalytic potentials of these thermophilic enzymes, and we would like to correlate both so that we can provide a good solution to the industries, because right now industries are needing some thermostable and robust enzymes which can work at very high temperature with good stability and good catalytic abilities. So we just try to correlate both the things. Today, I'll try to focus on molecular diversity aspects So as I told that we were interested in both culture dependent and metagenomic diversities, that is unculturable diversities, and we know that going for the culturing definitely leads to culture bias, right? And to avoid that, we try to correlate both culture dependent and metagenomic diversity. So it started with sample collection, followed by some conventional microbiological techniques. We also used ARDRA, that is 16S amplified 16S rDNA restriction analysis, followed by we went for the 16S rRNA sequencing of the selected isolates, and ultimately we went for the statistical analysis and stratification using the Clever software. As our focus was on diversity, we collected the samples during all the three seasons, and definitely bird season, while during monsoon only we received the thermotolerant bacteria from the you can say the proposed site. These all are the different biochemical characteristics and antibiotic sensitivity profiles. Like antibiotics were used to check the antimicrobial sensitivity profiles. Majority we used gram-positive. You can say the antibiotics which can act against gram-positive bacteria, because most of the isolates were gram-positive bacteria. They were true thermophilic. They did not grow at 37 degrees centigrade, which proved this thermophilic nature, as well as they all were spore forming. As far as the screening of biocatalytic potentials are concerned, majority of the isolates were amylase producers, followed by cellulase, while few of them, they could secrete both amylase as well as cellulase, while 12 isolates were lipase producers, but uniquely, we did not get any isolate which is protease producer. Our group is well known in the field of haloalkaliphilic proteases, but these thermophilic bacteria could not produce proteases, but the site can be referred as a marker site for the production of thermostable amylases. This is the, you can say the DNA isolation. We received all the isolates and we went for the DNA isolation from all the, you can say the thermophilic bacteria, followed by its amplification of 16 gene. This was actually followed by the ARDRA, where we used a tetracutter restriction enzyme. Especially we are using to get more and more, you can say, the genetic diversity. Here you can see that same 16S RNA gene, after treating with the HAE3, which is a tetracutter RE, we can get a range of these, you can say, the genetic profile, and based on which we can go for the stratification and the definitely you can say the genetic level, you can say the classification of these different bacteria. Clever is a software which is available, which is freely available online, and we use this Clever software to perform the in silico ARDRA. This is the results of in silico ARDRA. Here you can see the experimental, which I have already shown here. So this is the cuts and the fragment size, and based on which we could, you can say, the bacterial species, right? And these all are the sequences which we have submitted to ECBI. This is the phylogenetic tree. Okay, sir. Yes, sir. This is about the metagenomics. These all are this is about DGG profile, and this is the DGG.
Mr. Bhato, please conclude.
Like profile. Thank you. Yes, sir. Please. Sorry. If you have any questions, please, sir. Thank you. Thank you very much.
Any questions from audience and online person? Okay, thank you. Thank you. Mr. Bhato,
Yes, sir.
You just go.
Hello. Yeah.
May Jabin Sheikh, are you online? May Jabin Sheikh?
Yes, sir.
Yes, sir, I am here.
Okay, you start the presentation.
Okay, sir.
Thank you very much. Thank you.
Is it visible?
Yes, yes.
A very good afternoon to all of you. A very good afternoon to all of you. Myself, Mesbin Sheikh. I am a PhD scholar from Saurashtra University, Gujarat, and today I'm going to present on the comparative analysis on the molecular and metabolic diversity of marine actinomycetes. So the actinomycetes represent the source of tremendous secondary metabolites and bioactive compounds. So therefore, it is important to understand their diversity and taxonomy. So till date, only limited study has been carried out on the marine actinobacteria with respect to the seasonal variation, or we can say seasonal diversity. So this study has been explored the seasonal diversity and the biocatalytic potential of the actinomycetes from unexplored marine habitat. So first, seasonal marine actinomycetes were identified based on the 16S rRNA gene sequencing, and the season-wise phylogram were generated using the cluster W program of Megaten software. This is the offline software, and similarly the phenogram were generated using different kind of phenotypic character of the isolate, which are biochemical, different sugar utilization profiling, their antibiotic sensitivity profiling, their enzyme secretion, and this phenotypic data first converted into the binary matrix, and that binary matrix were used in the past software to construct the phenogram. So here the left side represent the phylogram, and the right side represent the phenogram of the winter season isolate. Here you can see here in the phenogram the axis represent the similarity level from 0.1 to 1, present the 10 to 100% similarity on the basis of the phenotypic characters. So if we compare the here the clusters were assigned at the 100% similarity level. So if we compare the phylogram to phenogram, if we see the one of the example like LW5 and LW2, which is the two different species of same genus Nocardiopsis, which is Desenvilla and Cinematophormus, they form the here they form the 100% similarity level. They form the cluster at the 100% similarity level, though they are from the different species. Another example is the different strain of Nocardiopsis alba like LW8, LW7 or LW9, which share the 100% similarity level in their phenogram. In the case of summer season, we can see here there are the less of phylogenetic diversity, but in the case of if we see the phenogram of the summer season isolate, here only the two clusters were formed at 100% similarity level. So they detect that they are having more phenotypic diversity. And if we see the example here, ALS1 and ALS2 share only at 40% or below the 40% see the phenotypic similarity. In the phylogram, it is proved that they are having they are identified as a two different species, like AL1 is identified as Nocardiopsis alba and ALS2 is identified as Cinematophormus. That is the reason why they are showing they are less phenotypic similar. In the case of monsoon season, only the two clusters were formed at the 100% similarity level, and different subclusters are formed below the 100% similarity level in the phenogram, which represent they are more phenotypic diverse among the seasons. If we see the example, if we see the example here, ALM1 and ALM2, they are belong to two different genus, which is Streptomyces and Nocardiopsis.
Has been.
Yes.
Your time is over. Please conclude.
Okay, sir. So overall, the on the basis of phylogenetic analysis, in the 3 seasons, they are belong to 3 different genera, majorly Nocardiopsis, Streptomyces, and Cephalopolyspora. And on the basis of the phenogram and phylogram comparison, the results on the the actinobacterial species have different clustering pattern and the different season. And overall, the study reflects that the significant phenotypic diversity of actinobacterial communities of the different season, despite they are having limited phylogenetic variations. These are the reference. I would like to thank my mentor, Professor S.P. Singh, for his valuable guidance. I would also like to thank Ministry of Earth Science Project, New Delhi, and UGC NFOBC Fellowship for financial support, and UGC CAS Department of Biosciences for infrastructure and other facility. Thank you.
Okay, thank you. So any questions from the audience? Those who are online, they can ask the question. Okay, thank you very much.
Okay, thank you, sir.
Mr. Dalip Rathod. Dalip Rathod. सर बाहर हो नहीं सर नहीं सर इसी कनेक्टेड नहीं
Hello, am I audible, sir? Hello, am I audible? Hello, am I audible?
Yes, you are audible.
Okay. So,
Dilip, are you listening?
yeah.
So please start your presentation. Time is very less, so we will get off. Dr. Rathod, we are not getting you online. So, okay, we are making you off. The next speaker is Ruhi Jan. Ruhi Jan, are you listening to me?
Hello?
Ruhi Jan,
Yes, sir. Yes, sir.
are you ready?
Yes, sir. I'm ready.
So you start your presentation.
Okay, sir. Sure.
Are you ready?
Yes, sir. Yes, sir.
Why are you making late?
Yes, sir. I'm.
जिस ऐप में खोल रही उसमें बहुत सारे ऐड आ रहे हैं सारे ऐड्स हैं जो खुल रहे हैं और बार-बार ओपन करनी बार-बार ऐड वो पीडीएफ ओपन ही कर रही है उसमें एडॉब में ऐड आ फिर ऐड यस सर नाउ
Hello? Am I audible?
Yes.
Okay, sir. Honorable dignitaries, esteemed scientists, Respect seniors, dearest friends, a very good afternoon. This is Ruhi Jan. I'm going to present biotechnology as a novel tool for biodiversity conservation. As we know, biodiversity, bio, that is life, diversity, that is variety. Biological diversity or biodiversity is that part of nature which includes the differences in genes among the individuals of a species, the variety and richness of all the plant and animal species at different scales in space, locally in a region in the country and the world, and various types of ecosystems, both terrestrial and aquatic, within a defined area. The word biodiversity originates from the Greek word bios, meaning life, and Latin word diversitas, meaning variety or difference. The whole word biodiversity generally therefore means variety of life. As we are dealing with biotechnology here, so biotechnology, it is the use of living system and organism to develop or make useful products or any technological application that uses biological systems, living organisms or derivatives thereof to make or modify products or process for specific use. Modern biotechnology began with the first recombinant DNA experiment but its real application started after 1980 when the US Supreme Court held that a genetically engineered microorganism was patentable. Now the biodiversity. Biodiversity is the variety of different types of life found on the earth and the variation within the species. So it is a variability among living organisms from all sources including inter-alia, terrestrial, marine and other aquatic ecosystems and the ecological complexes of which they are part. This includes diversity within the species, between species and of ecosystems. Now what is the role of biotechnology in biodiversity conservation? We all know molecular marker technology, additional marker, plant DNA bank, unconventional biotechnological methods, organogenesis, molecular farming, X2 conservation. These are all the roles of biotechnology in biodiversity conservation. Molecular marker theory, biochemical marker, phytochemical marker, DNA-based marker. Studies enzymes, variations and application in plant and animals. It studies about genetic variations within plants and animals. Phytochemical markers, they include the discovery of novel compounds from wild plant species and phytochemical markers discovery. It is an achievement towards enhancement of eradication of human disease. DNA-based markers are used in embryology, genetic engineering, physiology, taxonomy, etc. Additional markers. These are used for studying plant evolution, identifying of mutations by use of gene chips. Now the plant DNA bank. These have the potential for new resource characterization and utilization of biodiversity worldwide. DNA bank in Saudi Arabia is being constructed, which would be able to construct a genetic library, cloning of novel genes, so on and so forth. Unconventional biotechnological methods, micropropagation. We know it is an approach for conservation of plant species and one for preserve economically important crops. And this is used for production of virus-free plants and for in situ conservation.
Please listen me. Rohi, are you listening me?
Yes, sir. Yes, sir.
Please don't go like this. This is not classroom. You are sitting. You say what you have done.
Sure, sir. Sure, sir.
What you are doing. You are speaking like a class lecture. No, please say what actually you are planning, what you are doing, what you are actually doing in your lab. Have you done some work in these lines which you are saying?
Yes, sir. Yes, sir.
Say what you conclude that by saying that you have done these things. Hello?
Yes, sir. I'm listening, sir.
Yes, please say what you are explaining this is not the classroom. You have to say what you have done, what research you are doing. We are giving you one insight. If you have done something, okay, otherwise they will make loss.
Sir, I'm not getting, sir, clearly. Are you saying, sir, that I should not read like I'm...
No. I am saying you are explaining the things which are the basics of biodiversity and biotechnology. Okay?
Yes, sir. Yes, sir.
Listen these things. You know these things. What you have done actually, please say that.
So sorry, sir. I'm not getting, sir. What are you saying?
They are making law. Thank you.
Hello?
Guri, are you listening?
Yes, sir. Yes, sir, I'm listening, but
Make yourself quiet. Muthu, Sanjay Singh,
Yes, sir, I'm present, sir.
Muthu, Sanjay Singh.
Yes, sir, present. I'm present.
Okay, you please.
Hello, sir. Is my screen visible? Good. Good afternoon, one and all. Myself, Mukesh.
Sanjay, you haven't shared your screen.
The slide is not visible.
So I have chosen this one. I present the whole screen. So can I present it like this?
Sanjay, you haven't shared your screen yet.
Okay, sir. I just I will see. Okay. Once again, good afternoon, one and all who is present here. Their seniors, faculties, and my colleagues. Myself, Mutum Sanjoy Singh, and as part of this National Symposium Cum workshop, I'm presenting my work scope of vermicompost as an alternative mode for managing invasive alien plants. So these are a few glimpses of the invasion, and in this picture, it is this one, Mycenia mercantile and Azarethan conizoides invading a field site, and on the second picture is the Mycenia mercantile invasion on the bamboo beds. So coming to the introduction, so invasive alien plants, they are those species of the native origin, which one introduces to a new habitat, then threatens the new environment. So what we have done, and we have surveyed three states: Arunachal Pradesh, Manipur, and Nagaland. So and I've given the respective invasive alien plants recorded till now. So Manipur has got the highest 81, and Arunachal Pradesh has 61, and Nagaland having 37 invasive alien plants recorded till during our study. So successful establishment of these plants in Northeast India is resulted due to rapid increase in the rate of anthropogenic disturbances of our ecosystem, and And several attempts and methods have been made to control the establishment and invasion of this plant, either by destruction or utilization of the plant parts. So moving, these are the maps. First of all, it's for Arunachal Pradesh, and these are the points given where the selective invasive alien plants are reported. And for this one, and this is for the Manipur, and this is for the Nagaland. So coming to the introduction. So due to this invasion, so we have come to about this one about the management scope. So we have started this one. We have started looking for this management scope. So for this management, so we have chosen vermicomposting. So to what is vermicomposting? It is nothing but the excreta of earthworms, which is rich in humus and nutrients, and is greatly humidified through fragmentation of the plain organic materials by earthworm colonization by microorganisms, as given by Edwards Neuhauser. So for materials and methods, we have chosen 5 invasive alien plant species. These are given below: Ezeretina adenofora, Ezeretum, Comolena, Lentena, and Mycenia. The reason why we choose this plant species is that the species are this one frequently available, as as well as they are dominating in the in the nearby locations. And for the worm species, we have chosen Eisenia foetida because they have this one short life cycle, as well as they have high reproductive rates, and that's why they use in vermicomposting. So for the vermicomposting, we have set up a small composting unit for each of the species, and another control composting unit was also set up. So for these are the photographs of the species we have chosen. This is Egeretina idenophora. This is the Egeretum conizoides, and this one is Comolena odorata, which is also enlisted as the enlisted in the world's 100 highly invasive plant, and Mycenia migranta and Lentena camera. So this is the preparation of the experimental units that we have taken in the nursery. So these are the vermi beds. This fill up with cow dung, and this is the chop. This one invasive alien plant parts, and after that we have put in this bins and let it dry, and after that we introduce vermi this one Eisenia foetida in the this one plastic crates. So after that, we have done like pH determination and total nitrogen of the total nitrogen, total phosphorus, total potassium of the this one vermicast. So these are our results. So the concentrations of the first total nitrogen in percent. Total nitrogen was found highest highest in the Cromolena odorata of 1.83%, and this the control, that is of cow dung, was found to be 1.30, and for the rest 4 species were also found to be having higher concentration of total nitrogen than this one cow dung. And the lowest of this species was found in Mycenia migranta. This is 1.54%. And for the concentration of total phosphorus also, the control was found to be 1.14, and the rest of the species was for the rest, Ezeretina adenofora was found to be highest, 1.56%. Hello, sir. Sorry, okay, so just 1-2 slides. Okay, and so directly coming to the conclusions. So in our study, it has revealed that revealed a significant scope for this effective use of these plants as potential source of organic compost, which is rich in macronutrients through vermicomposting. So there is a need of identification of other potential invasive plants, which is highly effective for utilizing as raw material in this composting. So this type of potential utilization of this IABPS or for such value-added products may aid in effective control of invasive alien plants in this, especially in this in our Indian Himalayan region. And lastly, this composting of invasive species will also cut 60 to 70% cost of the fertilizer. And so finally, why vermicomposting? Because the process is easy one, and it is easy, and second, eco-friendly, and it can again it has economical benefits, as well as it can help in the regeneration control of the invasive species in our surroundings. So lastly, I would like to thank this one MOFCC for providing our financial assistance through the project entitled this Invasive Alien Plants in Himalayas: Status, Ecological Impact, and Management for carrying out the investigation. So thank you.
Any questions from any online or offline participants? Please feel free to ask.
Excuse me, sir.
Yes. Please, who is this?
Sir, I'm Sanjay speaking, sir.
Yeah. You want to say something? Hello.
Yes, sir.
Sanjay, you have shown some concentrations. These concentrations are of the leaves of the
Yes, sir. The voice is not. The voice is not clear. So.
Are you able to listen to me now?
Sir, I can listen you, but I cannot listen it very clearly, sir.
Okay, then we will stop here. I have some queries. If you are not able to wish you all the best. Thank you for your explanation.
Thank you, sir. Thank you. Oh, sir. Thank you, sir.
Sound कैसा है? Okay, so we have next participant, Mr. Dalip. Dalip Singh Rathore, are you ready?
Hello, ma'am. I am audible. Hello.
Yes. Please start your presentation, share your screen.
Okay.
Yeah, and try to be specific with the take home messages of the research.
Okay, the screen is visible.
Not yet.
Hello.
Now I think it is visible. Okay, myself Dilip Singh Rathore. And I am working as PhD fellow at the Department of Sciences, Saurashtra University. And Saurashtra University. Am I audible?
Yes.
Okay. My title of the presentation is Influence of the Oceanic Abiotic Factors on the seasonal diversity and richness of marine actinomycetes of the seawater of coastal Gujarat. So basically, this study covers the isolation of the actinomycetes from the marine actinomycetes, and this is the sampling site, and this part is a part of and this is a part of Ministry of Earth Science project, which was from 2014 to 19, and this is the glimpse of the study. Uh, the sampling from pre-monsoon, monsoon, and post-monsoon season. Then these were characterized according to isolation of the halophilic actinomycetes from intact sample processing, enriched sample processing, serial dilution techniques, calcium carbonate treatments, moist and dry heat treatment. Then we characterize by the morphological characterization, metabolic characterization, and molecular characterization. These are the physiological parameters, and from different seasons, and these are the parameters TDS and DO, salinity and conductivity. And this is the trend of isolation of marine actinomycetes, and the CFU count, and the number of the actinobacteria. As we can see that in sediment samples, the CFU count was high, and in water sample the CFU count was less. This is the graphical representation, and that's how the actinomycetes look like. And this is the seasonal comparison of isolates with respect to NaCl concentration. We can see that at the 5% salt concentration, which is about 1 molar of the NaCl concentration, the highest number of the actinomycetes have been observed. And this is the seasonal distribution of the morphotype based on the nature of the sample. As I discussed, in sediment samples, we receive maximum isolates. Then this is the morphological features on solid substrate. We can see that the majority of the isolates were rough in texture, and dotted appearance was there. And this is the shape of the colony, texture, elevation margin, and the pigments. This is the comparison of Cassidy isolates with respect to growth media. So we can see that in monsoon season, ISP 5 medium yielded maximum actinomycetes, and stars case in agar with the 37%. And in post monsoon season, there was a mixed trend of uh, all the isolation media. And we can see on the method by distribution, the intact sampling processing yielded maximum number of the isolates. The isolates were identified, and the phylogenetic tree were constructed. These are the list of the actinomycetes which were identified, and these are the accession number. Majority of the actinomycetes from the Nocardiopsis alba species, and Streptomyces species. And this is the trend that more distinct species in monsoon season as analysis and summary of
Summarize please.
okay, so the summary is the significant seasonal variation is there, and the influence of the abiotic factor is almost negligible, as majority of the actinomycetes are reported from the Nocardiopsis genus. So we can say that these actinomycetes are least affected. And these are my published articles, manuscript under preparations, and these are the achievements, and these are selected conference. Now at last I will acknowledge my guide, Professor S.P. Singh sir, Ministry of Other Science and Facilities and Infrastructure, created under various projects, UGC Department of Biosciences, and the LAM members. Thank you very much for your kind attention. Please, if any question.
Any questions from anyone in audience?
Please, any question?
Online or offline? Online participants may raise their hand or come directly to the question. No.
Hello, I think the audio is not clear, sir.
Okay, no problem. I think there is no question. be very well. Thank you very much for your presentation.
Thank you.
Mr. Chaurasia, Gajendra Chaurasia is there. Can you hear me, please? Gajendra Chaurasia, I call upon you. Gajendra Chaurasia. Gajendra. Hello. गजेंद्र, अगर आप सुन रहे हैं तो अपना माइक अनम्यूट कीजिए। आपकी आवाज नहीं आ रही है। गजेंद्र, can you hear me?
Yes, sir.
Okay. आप शुरू करें अपना प्रेजेंटेशन।
Yes, sir. Sir, my screen is visible. Good morning to all of you. This is Pratya Gupta, and my topic is evaluating physiological and biochemical responses of amaranthus fluentus toward polyaromatic hydrocarbon induces oxidative stress. And objective. Yes, sir. Your voice is not clear. Yes, sir. My screen is visible, sir. Yes, sir. Sir, can I start?
What I am saying.
Sir, can I start? Good afternoon to all of you again, and I Pragya Gupta, and am pharmacy third semester pharmacognosy, and I presenting on evaluating physiological and biochemical responses of amaranthus fluentus toward a polyaromatic hydrocarbon induces oxidative stress, and it have content objective, what we did, what we got, and research outcome. First, main objective of in this topic investigate the responses of dietary plant toward polyaromatic hydrocarbon pollution in then pollution for industrial exposure, like we see in this picture. 16 type of polyaromatic hydrocarbon affect the dietary plant, and people we consume that, and how to it affect their biological system. Then what we did, first we selected our plant amaranthus fluentus in the location of Tamnar industrial zone site, which are highly polluted area. Then our methodology is first we analyze histochemical analysis in leaf localization of reactive oxygen
reactive oxygen species, and second, our procedure is location of phenolic and flavonoid evaluation, and cell permeability check thought acridine orange UV fluorescence microscopy, and then we evaluate the nutraceutical profile. It have we doing total phenolic content procedure follow and DPPH radicals for antioxidant activity. Then what we got in histochemical analysis, first cell that evaluate. Cell that is visualized blue patches on leaf surface. Thought one blue chemicals. First non-polluted site it is or polluted site we you can see in this picture. Then NBT have cell that localization. Then H2O2 localization is you can see in this picture clearly how to it affect what is dry. Then what we got in histochemical analysis in staining method in acrid in orange solution. Acrid in orange solution have first we analysis control sample is parenchymatous all are seen clearly, but sample not clearly it total destroy. Then checking for phenols, it dipping on NPR reagent. Control sample is bright color is phenols, but sample has totally destroyed a little amount of present. Then total phenolic content in their result is not coming remaining, and antioxidant activity have there are standard curcumin, we take curcumin. Then control of sample have 56.4 and polluted sample is 11.8. Then we got nutraceutical profile for phenolic fingerprinting HPTLC. We take mobile phase toluene, ethyl acetate, formic acid. Then first picture is non-polluted. It have highly content phenolic. Then second picture is little calm because it is polluted site. Then our research outcome is depletion of nutrition pull more than 50% of plant growth, close victini of thermal power plant accumulation, and such battery plant which are grown in close victini to thermal power plant may not provide enough nutrition principle upon consumption, and cell also transfer in accumulated polyaromatic hydrocarbon in leaf tissue in human biological system. Antioxidant principles are more plant grown in close victini to thermal power plant when compared to control. Higher percentage of location is free radical was detected in edible plant grown in pH. Thank you, sir. Sir.
Yes, please.
Sir, trying to complete it. Can you?
Yes, please.
Can you ask any questions? Yes, sir.
Thank you for your presentation.
Yes, sir.
Okay. So this is is there any more question? Okay.
Hello, I'm audible to all of you. So next presentation will be very distinguished Professor Uma Shankar from Botany Department, Northeastern University, Shillong. He is the actually keynote speaker, but because of some engagement, could not deliver the keynote address in the morning. So now he is ready and he will begin his work. Professor Uma Shankar is well-known forest ecologist in the country. He has done his PhD from Northeastern University and he has worked extensively in the hotspots region of the biodiversity.
Sahu?
That is the Eastern Himalayas, which is stretched from Sikkim to Arunachal Pradesh. Then Western Ghats, yes, Western Ghats, BR Hills. So he is well known to the biodiversity. And today he is with us and he is going to share his experiences of his research work and how it can be helpful in evolving a center for biodiversity in our industry. That is the dream of our Honorable Vice Chancellor to have a center of biodiversity which can monitor, identify, and have people representative in the conservation of the biodiversity, particularly in this part of the country. Because central India is also very important landscape because it is in between the two hotspots of the biodiversity, that is the Eastern Himalayas and the Western Ghats. So now I invite Professor Uma Shankar to deliver his lecture.
Thank you very much, Professor Tiwari. Distinguished audience, online connectees. I'm blessed to be here amidst you to share my views on this very important topic of the seminar. And there is not much time. Of course, we are heading for lunch now. So I've been asked to share some of the summaries of my work or ideas. And that's what I will do. The title of my topic, which has been listed here, is Biodiversity in Mountains of Central Region of India, Inventory, Long-Term Monitoring, and Conservation. This entire organization of this particular meeting, seminar cum workshop, has been with the theme that we talk. Not going online? Okay. Okay, okay. Please share. Like, okay. So Central Indian region is a very, very important region from the point of view of biodiversity, and this has been the burning the burning issue for quite some time now, because if we look at the country level, we have been paying a lot of attention towards Western Ghats biodiversity in northeast India and Himalayan region. Somehow, the central highlands, which are there below the Gangetic plains, India, have been paid less attention, much less attention comparative to the other hotspots of biodiversity. So this is one of the things which has actually led the organization of this workshop, and it has been quite interesting since yesterday to learn the things, and I have been here and learning some of the things which we talk about, that what are the things which will be important for biodiversity enumeration for central India region, central highlands. Basically, this region comprises of 2 or 3 major mountain ranges we call Vindhyan region and the Satpura ranges. And these 2 ranges are, if we compare to the Himalayas, are much shorter in height. In Himalayas, where we have the highest mountains in the world, here the height of the mountains is below 1400 meters. Actually, Vindhyan region is below 800 meters, and Satpura region is something like around 1300 meters also. That's the maximum we reach. So what does it mean is that the climatic conditions in this region are much different from what we see in the Himalayan and northeastern region. And because of that, what we can expect is a change in the different kind of vegetation. And that is quite visible if we see the flora and the fauna in this region, particularly of dry deciduous and moist deciduous, little bit of moist deciduous, but mostly dry deciduous nature of the vegetation. It's mostly tropical in nature, and the tropical vegetation of dry deciduous nature here is tracking more to the Deccan Plateau rather than to the northeastern parts. But then, if we look at the Indian subcontinent in totality, then we find that there are similarities to some extent with what we get in tropical to subtropical areas of the northeastern region, with the central highlands also. Another very important geological feature of these hill ranges is that they are not of the Satpura and Vindhyan, as I have known. I am not expert geologically, but these are not of the tectonic movement result, right? So the collisions which have happened, the uplift that has happened in the Himalayan and northeastern region has caused intermingling of flora quite a bit. And here, because these are the older formations of these Himalayas, so flora looks like more primitive here in this region. That needs to be actually investigated and proved. Okay. Some of the researches very preliminary have been done in this section, but there is a lot to be done. And this is one area of research where we can really see wonderful results coming up in future. Another thing which I would like to say is that the plant functional traits of these species found here
are to be worked out in greater detail. And they may be quite different, okay, as a gamut, than what we see in the subtropical to temperate regions of Himalayas, and also subtropical to subtropical to tropical and subtropical regions of Western Ghats. So that is another area, and overall research on plant functional traits in India is not much. This is a deficient area. And here, of course, there are some groups like some groups from BHU and some other places have started research in this area. But not many people are working on plant functional traits. There is a lot of interest internationally. Now we see a very large group papers in Nature and Science on plant functional traits where they are placed. And these papers are coming up, and we need to contribute to this direction. And that is where I feel Central Highland can contribute a lot in this direction. Another very important feature I see for Central Highlands is that this region is also occupied by the tribal inhabitants native of this region, right? Like we have in several parts of Northeast India and in some parts of Himalayas and in Western Ghats. So there is a lot of native interaction between the people for their livelihoods and the natural resources in the forest. The sustainability of living of these people is again a very important area of research, wherein people derive a lot of non-timber forest products, both for their livelihoods as well as for some sort of sale to generate cash income. Okay, so these are some of the areas. Actually, if we go back a little bit, some 30 years ago in the history, not much, 30 years ago, most of us have been the students of biodiversity, and then we have been taught that there was something called biodiversity convention in 1992. There is a follow-up of the Earth Summit, right? And the term itself, at that time, we used to write biological diversity into words as a phrase. Now it is, and in dictionaries, if you see all the dictionaries of 80s and 90s and up to 2000 published, they will not have a term biodiversity. None of the dictionaries, English dictionaries. So that came only after, I think, 2005 or 6 dictionary as a single term. But then, what actually happened was, I have many slides, I'll not take much time. I'll go straight away to this particular thread of this. So, how to move slides? Can someone help me? I will come back later. No problem. So what has happened is, I will recall, please just excuse me. I recall that there was a paper published in 1988 in the journal called Nature, which is the top class journal, right? And that was on the evaluation of the vegetation of Amazon forest. This paper published in 1988 was a landmark paper. And landmark papers are those which change the direction of the research. And this was one of these. And this paper actually led the world to think afresh that what is the utility of the forest other than timber. Alright? Because forests historically, if we see in our country and in many other countries for that matter, have been thought of a source of timber, okay, major source of timber, and all other items extracted from the forest, most of them were left to the local inhabitants, like tribal people mostly, who were interacting that, okay, The major chunk timber as a commercial product is extracted by the foresters and rest is left to the people to survive on a sustainable basis. So that was some of the things which were happening at that time. So the new hypothesis in 1988 came that, okay, how much is the value of a hectare of the forest? Right? And this value was estimated not only in terms of the items we extract from the forest, that is called goods, but also services the forest provides to us. So services are innumerable and unparalleled and cannot be augmented easily by human intervention. So these services were valued truly, and with the value attached to the goods other than the timber. And this was very important turning point, and this led to a lot of policy generation. And when there was Earth Summit, then you will see that not only conservation of all forms of life, that is called biodiversity in the CBD document, but also their conservation and equitable sharing of the benefits. These were the three objectives of it. So equitable sharing of the benefits arising out of the biodiversity are to be ensured by the parties who are involved are signatories to this convention. India is one of those. And this is where the third one, equitable sharing of the resources, is of paramount importance to Central Highlands, Central Indian people. What I see here is that there are people who are partly or obligatory dependent on forest resources here. They have their cultural traditions descended for ages, and these cultural traditions are actually binding them together with nature in a very natural way. And these are to be documented properly. And the items and the services they are gathering from the forest are to be documented. Some work has been done, there is no doubt that we have to begin from the scratch. There is work which has been done, we have to build on that. So if something has been done, that does not mean that nothing more can be done. It means that a lot more can be done actually. And that is where we have to have conviction that, okay, if something has been done in some direction, that there is a lot of potential. And that's where I see Central Highlands have a lot of potential for future research. And this has to come to the black and white, and so that we can not only conserve biodiversity, but also add value to it. Value addition is another thing which can raise some cash generation, and improve the high livelihoods of the people here. Okay, so this lifestyle change can occur to some extent while conserving the resources. So this is one of the things. I have my own experiences to share with you. In 1992, I joined a project led by Professor K.S. Nada, who later founded A3 in Bangalore, ATRTE, this organization. And this founding of this organization was a far out of what we started doing in 1992. And we started doing, me and one of my colleagues, under this project, we started doing it before the Earth Summit this year. So it was 22nd February when the first draft, today is 22nd February, probably, I think. So this draft was ready for this biodiversity convention. And then sometime in June, 25th June probably, I think, if any of you remember that date. This was the meeting, and this came into being in 1993, December 26, I think. So this was effective, CBD was effective. Okay. We started working before this because Dr. Bawas, he was in USA, he's still in USA at the University of Massachusetts, Boston, considered as the brain of America, right? And he brought these ideas and he put them to work. I was a fresh PhD that time, and I moved from Shillong all the way to the district Mysore, right, in Karnataka, and we were put directly into the wilderness. We had a lot of all animals and all that was there, and without any production years. We used to walk kilometers every day and work right in the field without any production. Just with the help of two of our field guys, we have local tribal people, barefoot and with hardly any shirt on their body, but they were equipped with all the skills to survive in the forest. I was also young and had conviction that if the tiger comes, I will climb it. That time I did not know. That tiger also can climb the tree. Okay, that I see on National Geographic now that how easily a tiger can climb the tree. Those days I used to feel that I can climb the tree on the back of this going, then I will be safe. No, not anymore. So when those were days when you are young, you have a lot of energy and you don't think of any negative. Some point to. So that's how we worked 3 years continuously in the forest. What this work brought out was the first of its kind of work in India. And that was showing the impact of the harvest of non-timber forest products in the field by field work. Okay. We did 3 years of work and then we brought out some papers. And this was the first of its kind of work to show that if people harvest certain products, how the composition of the forest, the phytosociology term you must have heard of, okay, changes. How regeneration of the species changes in the forest. And this was very well taken by international audience. We got it published in a series of 6 in the journal called Economic Botany. And this has become a reference point. And becoming a reference point of your research should really please you to the core. Okay, and that's what happened. A lot of work started in this direction, not only in India, but also in many other countries, like wherever there was a lot of work. So that way we started. Then we stand today. We have a complete framework of biodiversity. Now CBD is there. We have COP meetings every 2 years organized. One of those were organized in Hyderabad also recently, 2 years ago. And the last one was very, very recent. Which place I don't remember exactly because it was online. And they keep amending the provisions there. But the basic tenet of this CBD remains the same, those 3 objectives I narrated. And I feel here in Central Highlands, the dependence of the local people or even people other than the tribals directly living either in the core or on the field of the forest, like we also depend on the forest products we buy from them, right? So our dependence is also there. We are not left behind. Some products at least we will see, right? So these forest products, how can they be sustained in longer term? And how can we add value to increase income of the people? So that was the hypothesis, which was the initial hypothesis when these kinds of work started in 1990s. But a lot of interventions from the economists, a lot of interventions from other biodiversity scientists, a lot of things. Initially, people started thinking of only plants, flowering plants, but then animal products are also there. They are also comprise biodiversity. Added. We know that there is a lot of illegal trafficking of many of the animal products, and that is to be counted. Traffic is one organization in India which is scouting that and working on those lines. So what I feel is here that the kind of work which has been done, value addition. So we gave one model of value addition from this particular work, and his wildlife sanctuary. These are the wildlife sanctuaries. And this was the first of its kind model on one of the species, which all of you know, that's called Phyllanthus indica amla.
वीडियो मतलब कौन सा है? ये वाला चाहो
So we gave this and how people were extracting amla from the forest, that I have actually documented there. Being with the Soligal, harvesting in the forest, seeing that how much percentage of the branches of a tree they harvest for the fruit. So we did a lot of work monitoring there in the forest, and then came out that can we find levels of sustainability with which that we can say that these many percentages of the fruits of a tree can be harvested, so that the regeneration of these trees in the nature is not affected and the future population is maintained. That was the objective, and this we did in the wildlife area. And this article was one of those in the series. So this was one model which was published. Another one I did was in Darjeeling Hills, Darjeeling area, and that was for one of the products what we call it Punjabu. What's the particular name of it? Isenolina maxima, but nowadays it is maxima. But this Jharu those days, I worked in 1995, 96 there. This used to cost some 5 to 7 rupees per piece. How much it is now? It's 100 rupees. Right? Exceeding 100 rupees. Those days when I used to tell that this will be sold in 50 rupees, people will laugh. What is it telling you? 5 rupees item. But now you see, you go to Amazon and Flipkart, it will be costing you something like 1 to 200 rupees. And there is a change now. You have plastic alternatives available, right? So these alternatives have come. Why? Because industry sees value in it. So they have devised plastic alternatives that, okay, this is one of the products. You will not believe that this particular Jaru, this plant used to come. If you ask your grandmas, you will tell that, okay, we did not use this in our time. Means before 1960, this was hardly used in areas other than where it was grown. But then gradually it started. In South, when I was working, people in South India did not know this. They used to use Jaru from this coconut ribs. Okay. But now you see everywhere it is used. All right. Times will change. This will also go down in next 10 to 20 years, hardly. Because alternatives are available right now, and cheaper alternatives. So cheaper alternatives will outweigh it, right? So that kind of thing. So this was one of the models which we see. So I found while working on this, most of the production of this particular item used to come from the tropical to subtropical belt of the hills of the Himalayan terrain in eastern Himalayas and also in Meghalaya. Meghalaya is one of the largest producers of this product. Darjeeling Hills, some Kuch, Bihar, and it is also coming from the hills in Nepal, imported. And from there, it is exported throughout the country and distributed throughout the country. And also exported in the Middle East countries, those days also, 25 years, and maybe now. So this is a very good example of a value addition model. Okay, this sustains lakhs of people and lot of weaving kind of artisans, you can say, people in different towns. So it comes raw from there. They will tie the Jaru in different ways in different towns. So it sustains people's livelihood. So this is a value addition model. So these are some of the stories. One more I will tell you is on Senamamum Kamala, This work also we did. You see, when you go to order a rice kebab in any of the hotels, there will be one leaf in the rice kebab. And that, without that, it's incomplete. right? And what is that? This is cinnamon Tamala. Meghalaya is one of the largest producers of cinnamon Tamala, which you use. Some comes from the Western Ghats also, but the economy of Meghalaya, if you see, among the top 3 natural products, cinnamon Tamala is the first one. Haru is Thysanolina, cinnamon Tamala. These are the top 2 products from there, even today. So, such is the importance of the non-timber forest products. I have this cinnamon Tamala is used leaf as a leaf. Generally, some people use it as a spice by grinding it. So, not many of the value addition models are there because it is used like that. 10 rupees per packet, some 10 to 12 leaves are there. I mean, if you go to a posh place, 1 leaf may cost you 2 rupees if you work down. How costly it becomes. Okay, so that's how there are many NTFPs. Now, the task is to explore the NTFPs here. Already lists are some lists are there. But to work out their quantum, to work out their sustainability in the long term, the harvest processes, can we have small interventions in the harvest process of the people right in the field so that it becomes more sustainable, just to teach them, just to train them? So that kind of intervention is required from the scientist side. So that can be done. And moreover, now there is one organization called TRIFED, TRIFED, by the government of India, which is provided a platform to sell NTFPs nationally from anywhere to anywhere. And this is one of its kind of the platform in India, which has been digital during last 5-6 years. And we should be proud of it. At least we were looking for some such platform. Now people can directly sell it to the. It's like Amazon for NTFPs. I can say that way to make it easier. Like Amazon for NTFPs. So anyone from anywhere can sell. But are people selling it that way? Not many, because there is a lack of awareness, lack of confidence among the people. Now we are in a situation wherein the smartphones have invaded each and every one, and we find even in most of the rural areas people are holding smartphones, right? So we can train them, we can make them aware, we can link them, because people mostly fear because of the small financial transaction. That fear is still there. Because somebody pay me by Paytm. No, I will not do something, will happen and all. So we have to, we have to train them. We have to bring them with confidence that they are okay. Apps have become so easy, just a few touches and then work is done. That can be done so that people can learn. Or at the ground level, panchayat level, and all, some sort of self-help groups can be made, and these can help these people to collect larger quantities from within a village and then sell it together and then distribute money among them. That kind of things are possible. Most of the demand is coming from the pharmacy industry for the medicinal line and spice industry. And third is dye industry. So these are some of the things wherein there is a lot of demand. Always demand is more than the supply. Am I right, sir? So these are the things wherein. So what has happened is that we can also explore the potential of cultivation of some of these items which are already in demand. When I meet sometimes the people from Imami, people from Dabur, people from Hundred, and all these people, they will always say, can you provide us a ton of this particular plant? We are 44 years, therefore I will claim I cannot as a scientist. But we can organize this to provide it. So some sort of this interlinkages to be established, market linkages to be established. They can be done here. Like you are in a belt where where this Lutea monosperma is grown. The flowers of which give you a dye, and this dye is quite rich and was used on a large scale in the past. If you look at the history of this dye of this Lutea monosperma, now also in demand, but we do not have, right? So this carpet industry requires a lot of dyes. There is a lot of demand. The carpets made out of the organic dyes are sold 100 times expensive than the normal ones in Western countries, but we are not able to supply. So these are some of the areas in which we have a lot of potential, and I'm very happy. So I'm very happy that the organization of this workshop and symposium has happened here with this primary objective, and Guru Ghasidas University has come forward to take this challenge up with their team, and with the agility of your new Vice Chancellor, I think things will move forward, and I am really inspired with the agility of Professor S.S. Singh, who has been untiringly working to organize this in a very, very short time. I did not expect, sir, I can tell you that you will have so many people connected, so many abstracts coming from different parts of the country, whole country almost covered, and people will be so interested here. But then this has been the reality. I think the abstract volume itself reflects that what kind of interest is there among the people in our younger generation, and we need to just foster it further so that people just put in there all their energies which they want to, and work for the good of not only of their academic pursuits, career, but also for the welfare of the people and upliftment of the nation. So that is the very, very purpose. This entire team of conveners, co-conveners, I mean, I cannot name all of them, but when I say team, everyone is involved, including all you students, all you research scholars, all of you have done wonderful work here in the last few days to make this happen in a bigger way than expected in this workshop, in this symposium. But the work does not stop here. This is just the beginning, and there is a lot more to be done. Okay, and the journey begins here actually, and the journey should not stop, because we can foresee that there will always be successions, there will always be people removing and then taking people taking places, but then work should not stop. This has to be a visualization for longer term rather than shorter term. So maybe this story continues for decades and decades until we achieve something. The overall objective is to make people happy, to ensure their livelihoods. Nobody should remain poor and die of hunger. Okay, that is the major objective, and that is where the role of LTFPs comes. I have intentionally not pressed one of the LTFPs, and that is the poor wood, which is the largest of LTFPs in the country. Right? So one of the solutions which our Honorable Prime Minister brought for this was Ujjwala Yojana, in which a lot of cylinders have been distributed. Right? I'm just mentioning. But that is not the complete solution. Still, there are people, either they are not using it, or they have not got it, or still they are resistant to change their traditions from fuel wood to the cylinder. Okay, so that kind of resistance also we get to see, cultural resistance we call it. So that will be there. We need to interact with the people. In fact, I am of the view that we should not take away the rituals and customs of the people just for the sake of modernity, or telling them that we will ensure your livelihood, you give up this. No. You keep it, but then try to be more accommodative with what we can do for the sustainability. So what are our objectives? One is to ensure sustainability of the ecosystems and their functions, and to ensure that nobody dies of hunger, nobody remains deprived of the health facility. Okay, medicinal plants, right? So these supplies should be there in adequate quantity. That is the overall objective. Overall objective is not that I publish a paper in impact factor 5 and then we have. Well, that will be something, but not all. Okay. Sir, if you permit me, I would like to stop here, and because of the time constraint. So thank you very much for inviting me to Rastas University. I say all the tea and share my views with you. And if you have any queries, any online or offline participants, I'm there. Thank you very much, everyone. Yes, madam.
Good afternoon, sir. I'm Anjali Dandekar, research scholar from the Department of Forestry. And thank you so much for your presentation. Sir, as you have mentioned, we have been a lot of work with tribes also. So when we are dealing with tribal people and visiting their territory, so it is very tedious to get knowledge, better data. So I want your experience, your tricks and tips in a short way.
Yeah, actually, when you work in the field, you face a lot of vagaries of this kind. Okay. This is where you earn the confidence of the people. This cannot happen that you go with a pad of paper and a pen in your hand, and then, okay, chalo bhai shuru ho jao, I am asking you questions, and then you answer. No, that will never happen. Even if it happens, you will never get what you want. In the highest sincerity or honesty. When I worked, it was 30 years ago, people were little bit less exposed that time to smart technology was not there, computers were not there, right? But now they are more literate, okay? So more alert, okay? But then I had impediments that now you can go with digital instruments with you and without their notice you can record them. I did not have this facility, right? So I have to depend on pen and paper. What we used to do is that, as I said, I lived right in the forest with the people of their clan. They used to take me, we used to sit there, play tea, have tea with them, and this kind of food there, spend a lot of time. They started recognizing me that this some foreign fellow has come to our territory, and I was a foreign fellow for them. Okay, because I knew Hindi and English, and they did not know either of them. So communication was our sign language, and that's how I learned their accent. But whenever somebody foreign comes to you, you are curious and also good, right? So that kind of fear, when it faded away, then only I could approach. So you have to spend time, earn their confidence, and then only and depend on the local people. There is a lot of expenditure involved in this process. Okay, because you have to live there. Your TA will be for 1 day, but you have to live for 3 days.
सर आपने सही कहा कि उनका कॉन्फिडेंस तो आपको बिल्ड करना ही पड़ेगा, लेकिन जब हम फॉरेस्ट में जाते हैं तो हमको ये देखना होगा कि फॉरेस्ट का जो लोग रहते हैं सब बहुत सारी चीजें उनको जंगल से मिल जाती है। आपको देखना ही है कि उनको जंगल से क्या नहीं मिल रहा है, उन चीजों को फोकस करिए, और वही अगर आप उनको प्रोवाइड करेंगे तो वो बहुत हैप्पीली एक्सेप्ट करेंगे आपको, और आप जो भी उनको नॉलेज है आपसे वो शेयर। मेरा खुद का एक्सपीरियंस, मैंने अरुणाचल में काम किया है ट्रेडिशनल नॉलेज पे। वहाँ पे जो ट्राइब्स थी उनको दो चीज, नमक सॉल्ट और केरोसिन, जो जंगल से नहीं मिलता था, वो अगर आप हम लोग लेके जाते थे पैकेट और उनको देते थे तो बहुत मतलब खुश होते थे। और सारी ट्रेडिशनल नॉलेज जो उनको डाई की उनकी थी, मैंने डाई बहुत एक्सपेंसिवली काम किया है, वो बहुत खुशी से बताते थे। तो आपको यही करना है कि आपको देखना है कि जंगल से उनको क्या नहीं मिल रहा है, वही जंगल वही चीज आप उनको प्रोवाइड करेंगे, दे विल बी योर फ्रेंड and they will happily accept. Thank you.
Thank you, Professor Tiwari. Actually, क्या होता है, when we go there, they expect a lot from us. Never go well suited. Your attire should be like them there. First of all, try to mingle with them. अपना शहरी पहनावा पहन के मत जाओ. Okay. Try to be like them. Adopt your food habits also like them. If you can communicate with the language, that is so much Suhaga. Okay. These are some of the work. Never promise them anything that you will be able to give them, because you will not be able to give them. Okay, they think that okay, somebody from outside has come, this government mercenary and will give us this and that, and we will be happy tomorrow. Nothing like that. You tell them that you are a student, you have come to learn from them. I will not be able to give you anything, but I will be able to help you some way or other. Okay. So no false promises. So these are some of the tips that you want. Thank you. Anyone else, please?
Thank you, Professor Rohan Mukherjee. He was moving the foundation on the speech. Professor Rohan Mukherjee looks simple, but he's not simple. He's full of knowledge. He had extensive both the God and the artist. He explained the things, but very much concern of forestry students and scholars. But he is the only person of forestry. Thank you very much, sir, for giving such a nice and very informative lecture. And again, I am glad for Thank you very much. So now we will the lunch break. So after half an hour, we will be here and will next. Thank you.